सही, बिल्कुल सही कहा सर।
मैं जापान में गया था तो उन्होंने मुझसे पूछा, तो मैंने कहा $1 है। तो मैंने कहा नहीं, हम लोग पैसे की परवाह नहीं, नोबल प्रोफेशन। तो उन्होंने बोला कि सारे प्रोफेशन नोबल होते हैं, बट इन योर कंट्री डॉक्टर्स आर ट्रीटेड वेरी शैबीली।
बिल्कुल ही उल्टा हो गया। सर ये आप ठीक है सर। सर हम तो, हमारा एक्सपीरियंस है जर्मनी में। वो एक बार ट्रेन में जा रहे थे तो वहां पे उन्होंने पूछा, तो बताया अब हम प्रोफेसर हैं। ज्यादा रिस्पेक्ट से उसने बात करी कि एज इफ आई एम टॉप ऑफ द वर्ल्ड।
हमसे भी ज्यादा पहले रशियन का हाल बुरा था।
यार हम तो जैसे चल रहे।
So, Dr. Pancholi, I guess our esteemed guest and panelists all have joined, so we are good to start with the conference proceedings and all.
Yes, sir. We are starting it shortly, sir.
Right.
Where are you, Dr. Nagaish, right now? You are in Delhi or somewhere else?
Sir, Delhi only.
Delhi, okay. I thought you will be there.
No. I thought of, but then I realized let me have this event virtually only, because anyhow we have to shift this physical event in virtual mode considering the current situation, and not sure what exactly the situation at that, like today onwards. So maybe that was one of the reason I thought of having this virtual event in a hybrid mode, rather, I can say. So the organizer and the key office bearers and all from the Ganpat University, they are joining physically, and majority of the students of the Ganpat University has also joined in a physical mode, and rest of the participants and esteemed speakers and guests are joining virtually, so that make it a hybrid event basically.
That's the best, good thing for this conference should be benefited to the students.
Yes. At the end, students should be benefited out of it.
Yeah.
Hello.
But anyway, I'm very nicely organized.
Thank you so much, sir. Thank you.
Hello, Upendra sir.
Yes.
Can you please rename? We have 3 unknown guests. Can you please rename them?
I'll just check and I'll rename, no issues.
Thank you, sir.
हाँ जी सर, मैंने कहा करें स्टार्ट। सारे लोग आ गए आपके यहाँ से भी जिनको ज्वाइन करना था? अच्छा, ठीक है, कोई बात नहीं। मतलब मैंने कहा बाकी तो सारे लोग आ गए हैं, है ना? अच्छा, हाँ बिल्कुल उस चीज को कर लेते हैं। जी, राइट।
नोट करा दीजिए हमको, एक और मीटिंग से निकले।
डॉक्टर गांगुली, अभी आप कहाँ पर हैं?
मैं दिल्ली में बैठा हुआ हूँ।
दिल्ली में? अच्छा।
I have a problem that I am in attending NABH board meeting which was set up, but उनसे मैंने छुट्टी ली हुई थी कि 3 बजे से मुझे 10 बजे छुट्टी दीजिए, तो वो तो हो गया, तो स्टार्ट करा दें काइंडली तो अच्छा रहेगा।
नमस्कार सर।
नमस्कार नमस्कार ब्रह्मेश्वर, क्या हाल चाल है?
ठीक है, सब बढ़िया। गांगुली सर नमस्कार।
मिश्रा जी नमस्ते।
नमस्कार। अरे वाह, सब लोग हैं यहाँ पे।
वेलकम मिश्रा सर।
थैंक यू। सबको प्रणाम।
खुश रहो खुश रहो।
उपेंद्र जी नमस्कार।
नमस्कार सर। नमस्कार नमस्कार सर।
जीएन सिंह कहाँ है, दिख नहीं रहा।
ये है तो यहाँ पे।
अच्छा अच्छा अच्छा,
जीएन सिंह का स्माइलिंग फेस है।
अभी दिख रहे हैं। यस
सर, आज बड़ी सौभाग्य की बात है कि सारे गुरु, सारे मित्र साथ के एक प्लेटफॉर्म पे देखने को मिल रहे हैं। ये नवरात्रि की सबसे बड़ी उपलब्धि हम लोग की है।
वेरी गुड। हैप्पी नवरात्रि टू एंड हैप्पी दुर्गा पूजा टू यू ऑल।
थैंक यू
चूंकि हम कलकत्ता के हैं तो हम लोग दुर्गा पूजा ही बोलते हैं।
गांगुली साहब तो हमारे उत्तर प्रदेश के हैं शुरू से ही।
लेकिन मेरा बर्थ प्लेस कलकत्ता है ना।
अच्छा अच्छा।
ये कोई हमें ना तो दुर्गा पूजा एंजॉय करने देता है ना नवरात्रि। बैक टू बैक लोगों ने मीटिंग ठोक रखा है। आई डोंट नो, मोदी जी से मना रहे हैं।
सर, इसीलिए WHO आपको इंसाइक्लोपीडिया ऑफ नॉलेज इन मेडिकल साइंसेस मानती है सर, पूरा विश्व मानता है इसीलिए।
कोई फायदा नहीं है।
हम लोग को तो है सर।
इस बार सोचे थे कि दुर्गा पूजा कलकत्ता में मनाएंगे, अपना चाइल्डहुड याद करेंगे, लेकिन हो नहीं पाया।
अच्छा।
अभी सर फ्लाइट फ्रिक्वेंटली चालू नहीं है शायद ना। इट्स ओनली फॉर स्पेशल केसेस।
उस सब का रूल्स एंड रेगुलेशन एवरी डे चेंज हो रहा है।
कल हाई कोर्ट ने काफी छूट दी हुई है।
अच्छा।
कोविड प्रोटोकॉल बहुत कुछ कर सकते हैं आपकी बात। भाई साहब ये स्टार्ट कराइए।
It is humble request to all guests. Please keep your camera on. All panelists, please, sir.
चक्रवर्ती साहब शुरू कराइए, मुझे नहीं तो निकलना पड़ेगा.
Sir, we are starting within a minute. Yes. Seconds.
Oh.
Good afternoon to all excellencies, distinguished delegates, ladies and gentlemen. A warm welcome to Chief Guest of the function, Padma Bhushan Dr. N.K. Ganguly sir, President and Patron-in-Chief Padma Shri Dr. Ganpat Bhai Patel, Pro-Chancellor and Director General Ganpat University Dr. Mahendra Sharma sir, Pro-Vice Chancellor and Executive Registrar Dr. Amit Patel, Executive Dean, Faculty of Pharmacy and Science, Dr. S.S. Pancholi, sir. Distinguished guest, Professor Dr. B. Mishra, sir. Dr. Y.K. Gupta, sir. Dr. G.N. Singh, sir. Dr. N.R. Sheth, sir. Dr. D.K. Jaishwal, sir. Professor Dr. Harvinder Popli, ma'am. Dr. Upendra Nagaich. Emeritus Professor, Ganpat University, Dr. Bhashwat Chakraborty, sir. Dr. Ajay Gupta, sir. Girishbhai Desai, sir, Dr. Satish Shardana. We welcome all invited guests from SPER, Executive Committee members of the conference and dear participants across various states of India. It gives us great pleasure and privilege to extend to you a very warm welcome on behalf of Ganpat University, Faculty of Pharmacy and Society of Pharmaceutical Education and Research, SPER, who has clubbed all of us for 10th Annual International Conference and Exhibition, SPER 2021, on the theme Innovations in Healthcare, Integrating Academia and Industry, to mark Silver Jubilee Year celebration of SKPCPER. This event is sponsored by Gujarat Council on Science and Technology, Guj Coast, and Department of Science and Technology, DST. Academia and industry are two important pillars of our economy. A robust collaboration between the two will augur well for our developing economy, enabling innovations in healthcare, growth in the education system, and producing an employment-ready workforce, with the object of encouraging the spirit of research and innovations by integrating academia and industry. SKPCPR feels proud to host 10th Annual International Conference and Exhibition SPER 2021. Light brings brightness with it and removes the darkness. Lighting the lamp is in a way an invocation of the waves of knowledge that are activated on stage. We request all our guests to move to the image of Ma Saraswati for lamp lighting, the Deep Jyot, and the audience to stand up at their places. कुंदेन्दु तुषार हार धवला या शुभ्रवस्त्रावृता या वीणा वरदण्डमण्डितकरा या श्वेतपद्मासना या ब्रह्माच्युत शङ्करं प्रभृतिभिः देवैः सदा वन्दिता क्षमां पातु सरस्वती भगवती निःशेषजायापहा. Thank you, Hima and Prutha. Thank you, everyone. Moving ahead, I would like to invite Dr. Mahendra Sharma, sir, Pro-Chancellor and Director General, Ganpat University, for welcome address. Please, sir.
Good afternoon, everyone. The dignitaries on the dais, the dignitaries online, and all the dear participants. We are really delighted that the Honorable Patron-in-Chief and President of Ganpat University, Ganpat Dada, has sent his wishes all the way from California in terms of his message. I spoke to him in the morning and he said that he wished the annual international conference and exhibition SPER 10th a very huge success in terms of knowledge assimilation and dissemination, and that the object behind the organizing of SPER 10th will be served in the entire deliberations that may happen. We are very much delighted that the eminent professors, luminaries like Dr. N.K. Ganguly ji, former Director General, Indian Council of Medical Research, is with us. Dr. N.R. Seth, Vice Chancellor of Gujarat Technological University. Dr. Y.K. Gupta, President, All India Institute of Medical Sciences, Bhopal. Dr. B. Mishra, National President, Society of Pharmaceutical Education and Research. Dr. Harvinder Popli, President, SPER Women Forum. Dr. J.N. Singh, former Drugs Controller General and Secretary cum Scientific Director, India Pharmacopoeia Commission. Dr. Baswat Chaturvedi ji, Chairman Scientific Committee. He is part of Ganpat University now. Dr. D.K. Jaiswal, Pharmaceutical Consultant. Dr. Upendra Nagji, Convener and National Secretary, Society of Pharmaceutical Education and Research. And our own Dr. S.S. Pancholi, Chairman, LOC and Executive Dean, Faculty of Pharmacy and Science, Ganpat University. Dr. P.U. Patel, members of organizing committee, faculty colleagues, and all the participants. At the outset, I am really thankful to Society of Pharmaceutical Education and Research for putting their faith in Ganpat University and organizing their 10th annual international conference and exhibition at Ganpat University under the aegis of Faculty of Pharmacy. So we are really thankful to the entire team of SPER for putting trust in us, and we hope that the magnitude and the outcome would be as per their expectations, and we will try to achieve the very objective of the establishment of this annual conference and exhibition. Having said so, the theme that we are talking about, which will be deliberated during this entire international conference and exhibition, is innovations in healthcare, integrating academia and industry. Probably you have touched upon an issue which is the most burning issue for Indian education since last decades. And probably after our Honorable Prime Minister Narendra Bhai took over in India, and the type of initiatives he started taking up, he tried to create a bridge between the university and industry. It was never so that ever anybody would have imagined that industries would originate from universities. It was an island situation. Universities and industries would independently work together. And today also, when we deal with a lot of industries, at Ganpat University, if I tell you, we have centers of excellences with American companies, with German companies, with Japanese companies, but we are struggling to get Indian companies because we are still to fight with the mindset that industry and academia can work together. So it's where it's a very important initiative, and it has to be deliberated at length, because universities inside industry and industry inside university have to be inseparable part. If we talk about any world-class university, there is a seamless integration between academia, research, and innovation. They are all integrated. And that only happens if industry is an equal participant in this holistic process. If that doesn't happen, this is not possible. So look at our Indian universities. I'm not talking about uh, the blessed ones like IITs, NITs, and IIMs, who have been blessed for over the decades with the type of resources. But a typical Indian university, whether it's with the government or is with the self-finance sector, you will find that new knowledge development has been very slow process. And today, when we are talking about global rankings, ratings, all of a sudden we say Indian universities are not producing relevant patents. We are not producing relevant research. So this has all of a sudden come. But when industry and academia work together, we get action research. We get real-time problems coming to the university, and with the help of the resources available at the industry, the possibility of finding solutions which are new to the, unlike what we have been doing was new to the university or new to the market, and it was not new to the world. When you talk about radical innovations, you talk about breakthrough innovations, when you bring things which are new to the world. Our former Prime Minister used to talk about, Atal Bihari Vajpayee, I have been talking about this statement. He said, the future of India lies in IT. And people interpreted it as information technology, but he meant was Indian talent. And we never thought about it. And we have to start believing in it, that Indian talent does not need to migrate to Western world or to greener pastures, where there is a seamless integration between universities and industry, where there are tremendous possibilities of doing and achieving something. We need those opportunities to accrue here. And in last 7 years, we are seeing a sea change with startup and incubation centers given to the universities. Make in India and Atmanirbhar Bharat initiatives coming to us in a very big way. Universities today are more empowered. They are getting a lot of resources through various channels to undertake radical innovations, not only work on incremental innovations. What has happened over the period of time is we have worked on incremental innovations, which was anyway part and parcel of continuous improvements. But today we can think about our radical innovation. What needs to be done when you are talking about bridging this gap between academia and industry is to reorient your teaching fraternity. You have to reorient them to understand what industry needs, and you need to reskill and upskill them for that interaction. So there is a need which is very much relevant today. So it has multifaceted benefits. Probably one day seminar would not be enough to cover anything and everything. So it needs a deep understanding on how this interaction can benefit 360 degrees. And probably our new education policy 2020, which talks about skilling, which is meaningful education, which is employment oriented education, which is entrepreneurship oriented education, is calling for universities to reorient their syllabuses. Today, if there is a provision called multiple entry and multiple exit, that exit is not that easy, which we talk about. So say 1 year, somebody studies with you and wants to move out, the curriculum has to be mapped with the qualification framework, which are endorsed by the industry, so that the person who moves out gets a job based on that skill. And at every level, that mapping is done, not only at national level, but at global standards. So then there is an inseparable part. Industry you have to talk to and you have to work together. And if we talk about healthcare sector, there is a perennial requirement on a continuous basis of the practices to be clubbed. And that's how we can. There is a concept called talent as a service, which is doing the round, and industries are talking to the university to reduce the time in which a man they hire becomes productive on the job. Normally, if a candidate is picked from a university, has a natural time of becoming productive, probably 6 months to 12 months. By the time he starts earning something for the company, But if the engagement happens, that productivity happens sooner than it can be imagined. And the skilled manpower is readily available. So we have to think drastically, radically, and a strong vision has to be put when we are talking about integrating academia and industry. It's easier said than done. It's very difficult to map the speed with which academia needs to run and work with the industry. We have at Ganpati University several such initiatives. At our own pharmacy college, we have a tablet and a fluid manufacturing unit, which is run by our alumni. So it's a thought process that has been implemented. We have a semi-automobile assembly line under Japan India Institute of Manufacturing. And you know, the best part is they have created a methodology that the timing of the start of the college is the same, the time of the start of the shift at Suzuki factory. The breaks are given at the same time when the breaks are given in Suzuki factory. And they eat the same food which is served in Suzuki factories. So they are preparing you thoroughly. So from day one, once you pass out, the man does not feel anything new. When he goes to the job, he's productive day one. He understands the job, and they have invested heavily. Everything, the technology has been transferred seamlessly to the university. That's what we are talking about. And a heavy engagement is needed. Both the institutions, industry and academia, have their own sets of quality resources, and they have to appreciate and they have to accept those resources. Industry have to change their thought process. They should not think that universities are 20 years behind us or 10 years behind us. They cannot cope up with us. So those thought processes also have to be shared. They should also come forward, handhold the academia, help them understand their shortcomings, their weak areas, and participate in removing them. And if that happens, the time is not far that the concept of industry inside university and university inside industry will happen, and that would be real transformation for our economy and our society as a whole. On this note, I welcome all the dignitaries who have assembled to address this key conference of Society of Pharmaceutical Education and Research, hosted by Ganpati University Faculty of Pharmacy, and all the participants who are here to ensure that they have certain takeaways. And I always feel that these conferences should not only act as a networking point, but there has to be certain actionable points which should come out and which should be worked on. And when the 11th conference is done, this is the 10th one, when the 11th is done, those outcomes of those conferences, what action points were decided and what has happened, be presented, audited, and worked on. This is how the entire process of knowledge assimilation and dissemination would become a good. Thank you.
Thank you, sir, for your motivational words. We have virtual presence of Dr. G.D. Gupta, sir. We welcome you, sir. Now, the first distinguished to honor us with his presence is Padma Bhushan, Dr. N.K. Ganguly, sir, former Director General, Indian Council of Medical Research, ICMR. Thank you, sir, for your presence. We are glad you are here today, and sir is getting late. He is here with us in between another meeting, and I consider it a great honor to welcome sir to address the gathering. Please, sir.
And audible?
Hello? There is some disturbance, sir. We cannot hear you properly. Hello? Ganguly, sir?
Can you know?
So there might be some problem. We cannot hear you properly.
I don't know what to do.
Yes, now it's clear, sir.
Okay, so without wasting
Your
time,
camera is off, sir.
So camera is
no
on.
problem.
So thank you very much, the Honorable Vice Chancellor, Dr. Chakravarti, all the honored speakers and guests in the audience. I'll be very brief. Actually, as the Honorable Vice Chancellor has said, the Indian academia is very, very innovative and good. And then the same way, Indian industry has done us proud. We now know whether it is the vaccine industry, both of them have become major world suppliers and have produced products of the world standard, quality standard. And many of their products are approved by the different funding agencies around the globe. However, in India, that mixture of academia and industry hasn't happened that well. They have gone through their own path and they have excelled, but that meeting point has been lacking. Recently, we saw that the products which the industries have brought out and put in the market, most of the products were either licensed by major companies or they obtained from various startups which are all around the world, or purchased the technology in creating a product. There are systems which we have put up in India which might make a difference, but these systems are very nicely entrenched globally. Like I give you an example, like the Swedish academic institute, whether it is Uppsala, whether it is Lund, whether it is Karolinska, Gothenburg, all of the professors there, and they develop a product up to an end, and then the industry comes into that and takes up that, because the large industry doesn't have appetite to really go for early discoveries. Early discoveries are done by academia and startups, and then the larger industry looks at that and then they take that on. In India, we don't have two very major structures. One is the HTA, Health Technology Assessment Platforms, which many countries have. The Australians are very good at that. And the second one is that they do not have the health tech transfer courses in India, because we think that you just negotiate with the technology with industry and it will be done. There might be a product, but that product might not be ready to go, because what happens that there are so many things which a product needs to come to a fruition, which will attract the industry to take that product and buy that product. In our industry, is not that risk. It is really risk averse. It is not. It doesn't take unnecessary risks in the market, and every product has a risk. So the health technology units, transfer units within the universities, they look at that those gaps, and then they fulfill those gaps so that industry can readily take it. So in India, we have some very good examples of such associations. We call them incubator. We have a lot of biotech park. We have pharmacy park. There is one which JN Singh has put up in Noida for devices. So these are coming up, but the best academy industry association incubations I have seen in 3 places. Indian Institute of Science, I have also seen it in KIT University in Bhubaneswar, and there are about 40 different industrial houses in that university and a strong association with them. And the third one is the Rajiv Gandhi Center for Biotechnology, which is there with the and their incubation is with the Kochi Biotech Park. Of course, we have CCAM, that is in the Bangalore corridor, and we have a Hyderabad corridor also, where Rich and others are facilitating such interaction. So we have some very good examples, but the major products, whatever we are seeing in the market, 90% of them have come from the industry initiatives only. There are some reasons why the international guys, like in America, they have a big act as well as there is a law in which everybody's interest is taken forward. Patent laws also we follow now, and our clinical development, etc., have also become very good. But they were stronger at one time. Activities also help into such interaction, and we need to really align our activities to be more promotional so that they can meet those aspirations. We have several major industry academia interaction platforms which have been created. These platforms are NIMITLY, that is the Millennium Technology Initiative, done by CSIR. Then CSIR had also floated some quick major thing like affordable health technology, etc. Then we have TDB, Technology Development Board, which actually helps in walking the last mile. They also fund for the building of activities also. We have SIBRI, some Small Industry Biotechnology Research Initiative, and we have also BIRAC, which now goes up to the end too. So we have several such major programs, and some of the banks like ICI, etc., also provide funding and financing. And some major houses like Tata Innovation Center also provide. We have an innovation council in India. They also do hand-holding and provide a lot of support. However, we need to really align our regulatory activity to be more promotional, more not taking any shortcuts, but really aligning itself with the international norms and be promotive as well as help the industry and academia to go together by reducing as many bureaucratic red tape as possible. We need also to make many of time the academia, when they have floated their own company and they have transfer technology, many times they have fallen into trouble also, because there has been problems because of the lack of some of the laws which were put up, like we had put up Bedol. The has fully entrenched rules and regulations, but it has been very difficult to really implement them. We have opened NIPERS, we have opened many such structures which will, and these days the medical engineering and other allied sciences have also become very, very close, and some like Kharagpur IIT has opened a medical college. The Kanpur is on the verge of doing that. We have, like with the MIT in Boston, we have Broad Institute, we have Harvard and others. So these interactions are very, very necessary for innovation to move forward. So with these words, I will again thank everyone to providing me this opportunity. We have some very illustrious people over here who have strong associations with each other in this area, and they will be able to give you more things. But out licensing has been one major form in which our industry has obtained licenses, like the hepatitis C drug, like the remdesivir, like Herceptin. There are a huge number, rituximab, huge number of these have been now the Regeneron antibodies to Cipla. All of these have come, but our industry has made us proud. It has produced HIV drugs to the entire world, and has transferred technology also to many other countries. The same thing has happened during the COVID days. We are on the verge of providing vaccine when half of the world hasn't received one single dose of vaccine, India might provide other countries ahead of others, these things. We have some very good corridors for such industry. Gujarat is one of them. We need the state governments to provide such patronages and others. Hyderabad is the second one. Bombay was, of course, all the time there. But there are some very new ones emerging, which are also very innovative and very supportive. So thanks again to everyone. I see the hall is full. It has been a pleasure to interact with you. Sorry that I'm leaving you in between, but I will take your leave. Thank you very much for being patient.
Thank you, sir. Thank you so much for very informative talk, and thank you so much for sparing your valuable time with us. It will be a great pleasure for us to listen to you again in our future events also, sir. On behalf of our institute and SPER, we would like to present you a statement of gratitude, sir. I request Dr. Pawan, sir, to present a certificate of gratitude, please, sir. I consider it a great honor to welcome Professor Dr. B. Mishra, President, Society of Pharmaceutical Education and Research, India, and former head, IIT BHU, Varanasi, India, for presidential address. Please, sir.
Good afternoon, everybody. I'm audible, I hope so.
Yes, sir.
Okay. Honorable eminent dignitaries on the dais and present online, Professor N.K. Ganguly, Dr. N.K. Ganguly, former Director General ICMR and Chief Guest of this function, we have just heard him. Honorable Dr. Ganpat I. Patel, Patron Chief and President, Ganpat University, Mehsana. Dr. Mahend Sharma ji, Pro Vice Chancellor and Director General, Ganpat University. Professor Y.K. Gupta, sir, President, All India Institute of Medical Sciences, Bhopal. Dr. G.N. Singh, my very close friend, former Drug Controller General of India and Secretary cum Scientific Director, Indian Pharmacopoeia Commission, Government of India, and presently advisor to the Uttar Pradesh government. Professor N.R. Seth, Vice Chancellor of Gujarat Technological University at the Ahmedabad. Dr. D.K. Jaiswal, my guru, as well as being he at present pharmaceutical consultant and former director, Farias Laboratories Inc., New York, USA. Dr. Harvinder Popli ji, President, SPER Women Forum and Director, School of Pharmaceutical Sciences at the Depsuru, New Delhi. Professor G.D. Gupta, former President, Society of Pharmaceutical Education and Research and Director, ISF College of Pharmacy, Moga. Dr. Upendra Nagaich, Secretary, Society of Pharmaceutical Education and Research and Professor at the MIT University, Noida. Professor S.S. Pancholi, Executive Dean, Faculty of Pharmacy, Ganpat University, and Principal Sri S.K. Patel College of Pharmaceutics and Research. My very good friend and classmate, Dr. Bhaswat Chakravarti. All the chairmen of scientific session for 2 days, today and tomorrow. All the learned speakers from India and abroad. All the panelists, respected delegates, participants, media persons, ladies and gentlemen. As the National President of Society of Pharmaceutical Education and Research, one of the co-organizers of this conference, it gives me immense pleasure to welcome you all and deliver presidential address during the inaugural ceremony of this 10th annual international conference and exhibition SPER 2021 being held today and tomorrow in joint collaboration of SPER and Faculty of Pharmacy, Ganpat University. Sponsored by the Gujarat Council of Science and Technology and Department of Science and Technology, Government of Gujarat. In a bid to set COVID-19 pandemic blues, we count our most obvious blessings, such as India becoming one of the countries with the youngest population in the world, and our highest ever Olympic medal haul and highest ever vaccination against COVID-19, straws in the wind that herald the dawn of India's golden era. The last few years have changed the world. We have experienced the most devastating natural disaster in the history. The coronavirus pandemic and its resultant lockdown badly affected all major sectors of the economy, but it has come as a boon in disguise to the Indian pharmaceutical sector. COVID-19 has provided tremendous opportunities in the pharmaceutical sector, especially for India, though some part of pharmaceutical business was affected as supply chain and import of active pharmaceutical ingredients, which certainly has given the opportunity even to many Indian pharmaceutical companies to come out producing a good number of now active pharmaceutical ingredients and making India self-reliant and self-sufficient. Similarly, COVID-19 pandemic has had a profound and sudden impact on the educational process in formal and informal learning environments. It is in the context that our organization, Society of Pharmaceutical Education and Research, commonly known as SPER, has a pivotal role to play as a non-profit organization with the purpose of bringing together a number of interested professionals to exchange their views at a common platform provided by the conferences like this. Like every year, the theme of our conferences keep on changing, and we try to cover every aspect of our societal needs through our annual two-day conference. Occurring at the most opportune time, this time SPER 2021 has the theme, as you know, innovations in healthcare, integrating academia and industry. It goes well with the dictum, today's science is tomorrow's technology, and day after tomorrow's reach out to the society. Building bridges of trust and reciprocity between the academia and the industry is the need of present knowledge era. This conference is being organized with an intent to discuss upon recent innovations and developments in the healthcare sector. There will be plenty of knowledge enhancing activities such as keynote talks, oral and poster presentations, demonstrating new innovative outcomes, and informations pertaining to the development of healthcare products and services. During the two-day conference, we will also be witnessing the ritual of award ceremony at the closing ceremony of this conference tomorrow, where we will felicitate eminent teachers, researchers, students for their contribution to pharmacy, the profession, and also to the society. The main outcome of this conference is to impart a scientific basis for the integration of industry and academia. I am sure that the exchange of ideas in the relevant areas at the conference will prove valuable to find novel insights to solve persistent challenges in pharmaceutical domain. We hope this two-day conference would help all the delegates and students in particular to expand their mental horizon and would make use of this to the best of their abilities in future years to come. At the end, without taking our much time, I appreciate the dedicated efforts put by the organizers, especially Dr. Nagaich and Dr. Pancholi, and the members of various committees for meticulously planning an event of this magnitude and eminence. And I take this opportunity to thank you all for giving me this wonderful opportunity for presenting my views in front of you and for your patient listening to my views. I wish that SPER 2021 a grand success. Jai Hind, Jai Bharat. Thank you so much.
Thank you, sir. Thank you so much for sharing nice words with us. On behalf of our institute and SPER, we would like to present you a statement of gratitude.
Thank you. Thank you so much.
Thank you for your presence, sir. We have a virtual presence of Dr. Charanjit Singh with us. We welcome you, sir.
Thank you so much.
Thank you,
Extremely happy to be here today among the very learned group. So thank you so much.
The theme of conference attracts the focus of scientific community to resolve the issues and bridge the gap between industry and higher education institutes to prepare the next generation for challenges of future. With these words, I would like to invite Dr. G.N. Singh, sir, former Drugs Controller General and Secretary cum Scientific Director, Indian Pharmacopoeia Commission, Government of India, to say a few words. Please, sir.
Good afternoon, ladies and gentlemen. It's a great pleasure for me to see all the galaxy of my teachers, my colleagues, students who are really the backbone for making India as a pharmacy of the world. As I congratulate the Society of Pharmaceutical Education and Research, which is organizing this conference in collaboration with the Faculty of Pharmacy, Ganpat University, which is celebrating its silver jubilee year, Gujarat Council, and the theme which you people have chosen is really very appropriate and relevant to the pharma sector and health sector. It was my burning desire when I was serving through various platforms to see that the academia, the national laboratories, and the industry may join hands together and we may make successful stories. We succeeded partially, but now it requires to be accelerated. In light of the current test, when my guru, Professor Ganguly, my friend Dr. B. Misra ji nicely narrated, and my another guru, Professor Y.K. Gupta ji, would be speaking. I am seeing Dr. Sanjeev ji, who is always a man who made the India to stand taller when the whole world is facing to the challenges of the standards. These are the people who stands, and Professor Sanjeev ji, you were the man who made the things to possible. We innovated new pathway, how India can look different, but when there is a issue of not compromising on the safety of the people, he narrated with the collaboration with the industry and presented to the world that no, the academic institution, the standard setting platform, and the academia could work together. And such a successful stories are with us, the case studies are here. Now those things are to be followed in near future. Friends, I'm very delighted to see the galaxy of people over here. When I am talking, Dr. Professor Copley, D.K. Jaswal, who taught me, B.S. Chakrabarti ji, who is always close to my friend, and I am standing here today by his invitation. I am equally very delighted to see Dr. Upendra, who is doing such a wonderful job, Dr. Pacholi, and really the first conference on pharmaceutical education research was organized in 1988 at Singapore and Thailand. I was fortunate enough to attend over there, and I remember how the beneficial, the work which we are doing today is going to make a roadmap for success of tomorrow. Friends, I don't want to take too much of your time, but I appeal the young mind who is going to enter into the innovative phase. We are known as the pharmacy of the world. we are producing high quality drugs with affordable price. That is generic in nature. We are in the value terms, we are the 10th in the global scale, 4th in terms of volume. If you want to be the number 1 or 2 at the global level, then we have to enter into the innovative phase. And innovation would take place when we join hands together, as Professor Ganguly rightly mentioned, the Honorable Pro-Chancellor Dr. Mahendra Sharma ji rightly put up. This is the time when we have to follow the current government policy, when we want to see that India becomes self-reliant, either it is API, medical devices, or any other innovation. And Gujarat in particular is very progressive state. Most of the things happened over there. When I was DCGI, we were looking how the medical devices issue would be addressed. I happened to visit Surat, and I was excited to see so many good things are happening over there. Those things are going to be translated into reality, and the government of UP is going to establish a pharma park at Lalitpur, one medical devices park at Jewar, consisting of about 350 acres lands, and we are going to create state-of-arts hub in UP under the dynamic leadership of the Honorable CM Yogi ji, with overall support of the Honorable our Prime Minister, which is very closely and respectably known in Uttar Pradesh, and we are very fortunate and grateful to the people of Gujarat who have given such a wonderful person. With these words, I express my gratitude, but one thing I want to mention over here, the great vision of Padma Shri Ganpati Bhai Patel ji, who thought that this Ganpati University requires to be a pioneer institution in education, in serving the society. And we are surveying, you are at California, but we express our gratitude to you for creating such a wonderful facility platform so that such great people are here. Again, I'm very thankful to all of you. Namaste.
Thank you, sir, for your presence and words of wisdom. Thank you. On behalf of our institute and SPER, we would like to present you a statement of gratitude. Thank you, sir. Now let us have the pleasure of listening to Dr. N.R. Sheth, Vice Chancellor, Gujarat Technological University, Ahmedabad. I request sir to address the gathering. Please, sir.
Namaskar, I am audible?
Yes, sir.
Good afternoon to all the esteemed panelists, all dignitaries, Dr. V. Mishra, Dr. Y.K. Gupta, Dr. N.K. Ganguly, Dr. G.N. Singh, Dr. Jaiswal, Dr. Havinder Popli, Dr. Upendra, the Padma Shri, Dr. Ganpat Bhai, Principal, Dr. Pancholi sir, and all esteemed speakers and those who join virtually in this important function. So very first, I congratulate the organizer, particularly the Ganpati University Faculty of Pharmacy and Society of Pharmaceutical Education and Research for this important function, and the topic selected is very, very important. So all esteemed speakers, former speakers, they have focused on the academia and industry interaction. So it's a very, very important issue. But along with that, the innovation in healthcare, that is also an important topic. So we have discussed about the interaction, but as far as the innovation in healthcare is concerned, so very first, the innovation part. So actually the educational institutes, most of the educational institutes, the ecosystem for innovation and startup is not developed, because our education system, no doubt, the National Education Policy 2020, they have focused all such problems, and it will be in due course of time. Our educational institute will be a knowledge center. It will be a destination for knowledge from all corners of the world. But at present, our education system focus on education system, and the examination, that is the main focus point of the faculties, students also. So if any students is approach the teacher for any innovative idea, for creativity, but what the teacher says, he says, okay, your idea is very good, but you do all this thing after getting your degree. You focus in your exam, pass the exam with good marks, and later on you do whichever you like. Means we are killing the idea, we are killing the creativity at the educational institutes. So this is the in most of the educational institute, the situation like this. No doubt, by the initiative of our Prime Minister, Honorable Narendra Bhai Modi, now all institutes are planning for the incubation, for innovation clubs, and everything are slowly getting momentum. The Atal Incubation Center established at the various universities, various institutes, even the tinkering lab at the various schools. So no doubt, slowly the innovation and startup is gaining momentum at the various educational institute. But every educational institute has to create end-to-end support system for the students' ideas, for their creativity. And definitely, when such ecosystem will be developed, there will be a lot of innovation. Even during the pandemic time, we have seen, particularly in the healthcare system, there is a lot of innovation, because our young budding students, young budding scientists, they are capable to do when the challenge is given to them. So if there is a challenge, there is no PPT kit, no, there is a this oxygen. So number of scientists, number of institutes, they have worked on that, and the number of innovative products are in the market. Notice, you see, the PPE kit is not manufactured in India. The ventilator is not manufactured in India. But a lot of companies come out with their innovative ideas, and now every day we are manufacturing the 5 lakhs number of the PPE kit, and we are exporting. See the vaccine. We are in a surplus way, means 90 crores, more than 90 crore people, they have taken the first jab of the vaccine, and more than 30 crores, they have taken the 2 doses of vaccine. So it's a very tremendous work we have done in the healthcare system, but we have to do lot of work in the healthcare system. So in last decade, India has been witnessing an upsurge of innovation by almost all relevant stakeholders of the healthcare industry, including the area of medical equipment and devices, vaccines, medicine, medical infrastructure, diagnosis and service delivery, treatment of chronic disease, and launch of the patented products. So the academia will play a major role in healthcare sector by creating a culture of innovation, assessing the funding for innovation, addressing some geographical issues. The innovation, startup, entrepreneurship culture in the healthcare sector can become the bridge between the academia and industry. I think there are many more eminent personality here, and I am conclude here. Thank you for giving me an opportunity to interact with the all participants. Thank you. Thank you very much.
Thank you, sir, for your influential message. Thank you so much. On behalf of our institute and SPER, we would like to present you a statement of gratitude. Thank you again, sir.
Thank you. Thank you.
We have virtual presence of Dr. Indrajit Singhvi, sir. We would like to welcome you, sir.
Thanks a lot, ma'am.
This conference and exhibition has been conceived on the relevant theme at an appropriate time and in line with the National Education Policy NEP 2020 and during the glorious 25th year of establishment of Pharmacy College of Ganpat University. May I request Dr. D.K. Jaiswal, sir, Pharmaceutical Consultant, Jaiswal DMRK, New York, USA, and former Director, Forest Laboratories, New York, USA, to say a few words on this occasion. Please, sir. Jaiswal, sir. You're mute, sir.
Now it is okay?
Yes, sir.
A good afternoon to you all, and good morning to me. And thanks to Dr. Nagais for giving me an opportunity to speak in front of you all. And we listened a lot from Dr. Ganguly, just now Vice Chancellor Dr. Sheth, Professor Dr. Mishra, and Dr. G.N. Singh. They told so many things, very informative informations. Since, as you know, I am the US pharmaceutical industry experience for more than 3 decades, I would like to give some feedback to the students only. And just the pharmaceutical industry, just I would like to say, pharmacy is a very noble and honest profession. And in addition, it is very highly regulated industry. And I want to tell to all the students, never ever criticize if they will join to any pharmaceutical industry. Many people I notice based on my experience, that any new company or any new person join, and they always criticize the previous management. I just to advise them not to criticize previous management. The company has hired you to bring some constructive work, and something is missing, you can create and you can fix the problem instead of criticizing. In USA, that let me tell you, in the pharmaceutical industry, if somebody is criticizing, the management will show you the door. That's why never ever criticize. And the other things I want to tell, you never underestimate yourselves. Because sometimes some people got very underestimate. Every individual has got the, what you call capable of some strength. And pharmaceutical industry is not only R&D, they've got so many departments, quality control, quality assurance, regulatory, CMC sections, medical group. Do you have to work just like as a team? As team means together, everyone achieve more. And other things to get the work done. The bottom line, we have to cooperate. We have to, interactions with the each and department, and your objective should be to get the product out. And let me tell you one more thing. During this COVID crisis, I'm not going to criticize anyone here. Doctors are very good to diagnose the disease. But if we pharmacists is not going to produce any medicine, doctor can't do anything. That's why our role is very important. And we pharmacists train doctors and we tell them, these are the correct dose, these are the interactions with the other drugs, so that the pharmacists played a very vital role in our health consciousness, not only doctors. And also, I also appreciate engineers also. If engineers are not going to make any machines, then we cannot produce any tablet, capsules, and other injectables. That's why that is a teamwork. I always mention that one. And the other things, if you choose your career in pharmaceutical industry, the 7 very basic points you always keep in mind to remember. First, less problem in manufacturing. While working in the industry, we have to very well in advance, there's less manufacturing, less problems in manufacturing. We all need to ensure better design of products from the very early stage in order to avoid problems in manufacturing. And second thing, implementation of new technology. Always be prepared to implement new technology if needed in order to improve. Documentations, all documents should be very crisp and clear. Suppose any inspector is coming from FDA or any from the Indian delegates, you cannot fight with them. You have to convince them. And first try to find it out what is the person's background. If it's a science background, you can convince based on science. If it's not science background, you have to convince in a very simple language so that they can understand and they can approve your products. And other things is documentation. In pharmaceutical industry, documentation is very important. That should be very crisp and clear. Documentation plays a vital role in pharmaceutical industry. Proper documentations ensure less hassles during review. As a result, reduce deficiency and quick approval. And CGMP, as you already know, I have nothing to say, all the pharmaceutical industry stick to CGMP, and make a habit to routine follow-up and corrective actions on daily basis. And we have to cooperate with each department, with either medical group, either regulatory group, either quality control, quality assurance. You have to work together. You have to convince, otherwise you cannot get your product out in a very timely manner. And the quality assurance, as I mentioned, all clinical packaging, all the things, this should be released under the, what you'll call, with the quality assurance. And as I mentioned, the pharmacy is a living subject in which many important changes may take place during a single year. Therefore, it is entirely necessary to be in touch regarding new developments on regular basis. These activities will ascertain that the drug can safely be used in initial clinical testing, that can be assayed for, and that it can be formulated into titles that would be stable at least during the period of clinical testing and further toxicity testing. And let me tell you one more thing. Some people, they are always telling brand is also very expensive. Let me tell you the difference between brand and generic. Generic can make the product when the brand, what you'll call patent expires, they can make the product and they can sell the product. But brand name is expensive due to any reason, due to some reason, because any pharmaceutical industries like Pfizer, Merck, Glaxo, they are working at least about 10 and 12 products in the market at the same time. And after 10 or 15 years or 20 years of research, they got one product approval. And that's why for the other products expenses, they bear on that one because they are also expending a lot of money on the other products. And the brand name company got very fortunate if they get one product approval. And that's why they got the patent based on their research and all those things. And so that the brand name is expensive. Thank you all, because the time is very short. It's not possible for me to explain the whole experience in a very short period of time. If I will get an opportunity, I will visit your university and share more information. Thank you.
Thank you, sir, for sharing wonderful information with us. On behalf of our institute and SPER, we would like to present you a statement of gratitude.
Thank you.
Thank you, sir. Society of Pharmaceutical Education and Research, SPER, is a scientific, educational, and professional non-profit organization committed to promote the highest professional and ethical standards of pharmacy and to work for the growth, development, and betterment in the field of pharmacy. It is one of the leading pharmaceutical associations in the country and working in the interest of pharmaceutical professionals with a member base of around 3,500. It's spread across country and have 13 state branches. On this note, I would like to invite Dr. Upendra Nagait, sir, Secretary, Society of Pharmaceutical Education and Research, SPER India, to brief on annual report of SPER. Please, sir.
A very good evening to all. First of all, at the outset, I would like to extend my heartiest thanks for the organizer for having a collaboration with SPER for organizing this Decadian conference of SPER. It's a proud moment, indeed, being a national secretary, and it's an emotional moment as well for me. It has been the year 2010 when very like-minded people like all the presidents and office bearers of SPER, because we are very fortunate enough to have people, those have been the part and parcel of the society from the day one, from the scratch. When we started, we thought of given a thought to establish some sort of a organization to inculcate the highest level of education and research. And we started the establishment of society along with the establishment of a research journal with a name and style of JAPTR, Journal of Advanced Pharmaceutical Technology and Research. This journal is the official research publication of our society. Currently, this journal has been published by Wolters Kluwer, Medno, Philadelphia, US, and it has been indexed in Web of Science. From the very first volume, first issue, we are included in PubMed, Scopus, and all other indexing agencies. So indeed, it's a proud moment for society for extending the support from the members for inculcating the research, highest level of education and research. We have grown like anything in the past 10 years. We started from scratch, we started from zero, and currently we have our roots deep penetrated across different states of India. Currently, we have more than 3,500 life members across India and overseas with a strong presence in 13 states. So once I'm mentioning the 13 states, we have a state branch in those states. We have office bearers. Those are working, excelling as in part and parcel of the society and to maintain the level of the standards for pharma education. That could be the events that we are going to organize. We have organized many events in the past in terms of the academic and research endeavors. There are various other events that has been conducted under the aegis of SPER to disseminate the knowledge. And that was one of the reasons. Currently, this conference has been organized in collaboration with Ganpat University by the sincere efforts of Dr. Bhaswat Chakravarti, the current president for Gujarat State Branch SPER, Professor Dr. Pancholi sir, and all the other esteemed members of the university academics and administration who has supported this event like anything. So giving an annual report in the event of annual conference, and that is a decade conference, 10th conference, it's indeed a proud moment. We announced this conference to be a physical event, but considering the challenges that are going on with the COVID impact, we anticipated that might be the situation would be better, but we are not sure at that time. So we have taken a option of conducting the event as a hybrid mode as well, where we will be able to have the people who will be able to join the conference via physical mode. They will be able to join. They have joined, rather, I can say. I was able to see many of the students and the entire convention hall. The hall was completely full from the students. So that showed their commitment for getting some knowledge, getting some information from the esteemed speakers, panelists. Those have been the part and parcel of the society, and those have been the part of this wonderful event. It was very rare to see such luminaries of pharma community, healthcare community at one stage, and we are always very fortunate to have such people with us in one or the other event. I still remember the fond memories of the SPER collaborative event with Springer Nature, where we had the as a speaker, Professor Saranjit Singh, Dr. Mishra being a part of the SPER, being a national president, then many other office bearers and speakers for different events. talking about the various events that has been conducted by the society. So it has been the SPER Student Congress, that was the virtual event that has been organized by the SPER Student Forum. Because what we have inculcated in a way that our society, if we have one society, we are maintaining that standards, we are working for contributing for the society, then definitely we have to have all the verticals open with us. Saying this, we have our women forum that has been headed by Professor Dr. Harvinder Popli, being a national president of SPER Women Forum. We have our established SPER Student Forum that is currently being headed by Mohammad Ashar from Jamia Hamdard. He is currently the national president for SPER Student Forum. We created this forum ultimately to benefit our students, to benefit our young buddies, because somehow we realize that they have certain expectation, they have certain anticipation from their academic institution, from their organization where they have been studying in undergraduate, postgraduate, or doctorate level. But yes, there has to be some forum that is ultimately going to support them, going to address their issues, their challenges at national level, and provide them a path to work upon, to path to showcase their talent, to showcase their temperament, to showcase their own capabilities. And by virtue of, because everyone is busy in saying they are the real future, they are the backbone of any nation. But at the same time, we have to provide certain forum, certain platform for them where they should not feel offended. They should not a feeling that whether if I'll going to speak something, how the people are going to judge. We are not here to judge anyone, and we are no one to judge anyone. We are here to support our young buddies. Those are the young future. Those are the young brigade of any nation and a backbone. So that event was basically organized by Student Council, Student Forum, State and Central, and it has been conducted and everything was managed by students only. And we are very fortunate on that event. The chief guest was Dr. GN Singh Saab only, and then different other people from the industry. I still remember the fond memories when Mr. PK Datta, the founder and managing director for Cystopic Laboratories, has witnessed and graced the occasion with his presence, and different other people from the industry, PSD Chamber of Commerce being in part and parcel of the event. So that was one of the greatest event that have organized under the aegis of SPER and by SPER Student Forum. Last summers, when the lockdown was there, everyone was worried about their internship, worried about their training, how they are going to get the skill set, how the skill development would have been there, because everything was shut down. Neither they are able to visit any institute, any research lab, or any other industry for conducting or carrying out any of the internship program, training program. Also, realizing this fact, with the approval and kind support of the competent authorities and the office bearer Central Executive Council of SPER, we have announced an internship program for our students. Either they are a member of society or they are not the member of the society, but ultimately they have a zeal to work for the society, for the industry, and they wanted to learn something that is much and more important for us. So that event we have organized in collaboration with a market research organization to provide them a forum where they'll be able to learn the skills, they'll be able to learn the actual criticality of the industry, how industry is moving ahead, how industry is working. So giving that thought to the students, we have organized that industry internship program for the students for 3 months, so that students have enrolled, and there was nothing that any sort of a fee or something we have charged. Rather, that was completely entirely complimentary basis for the students, because we are considering not-for-profit society, we are nowhere engaged in money making or getting any sort of a registration fee for providing them the services or facilities or that kind of a forum to learn and get some skills from the industry. So that 3-month industry internship has been given for the students to basically identify the market research concepts and understanding the actuality, gain the practical experience from the industry. We also organized one of the event in collaboration with Women Forum, Student Forum, and different industry as an industry defined problems, where we have given a platform for the students to get understanding about their skills and knowledge, what knowledge they do have and how they are going to enhance their knowledge. That basically based on the pharma management subjects, integration of pharma and management, and that event was a great success in the history of SPER for our virtual events. Then different other light events we have organized as a photography contest, article writing competition that has been keep student updated, engaged, and at least the situation was such in the pandemic situation that everyone was so negative, so hopeless. And we as a society, it's not just our responsibility, this is our duty as well to get them aware, to get them motivated. Organizing an event is not only the purpose, giving them a focus, giving them a path is also very important, and keep them updated from the day-to-day activities. So that was one of the reason we try to involve the students to keep them engaged with the current happenings. And during the pandemic, that was very much required, because somehow they felt something that what we are going to, they understood that the dicey situation would be there. So for that, we organized some of the events where they are able to showcase their talent in terms of photography, article writing, essay writing, or likewise. So that was one of the event that we have organized. And then there are different other events that we have conducted in collaboration with SPER and Springer Nature. That was one of its kind of an event where we have collaborated with Springer Nature, one of the globally acclaimed research publisher across the globe for science, technology, and management domain. So they have collaborated with SPER, and that was one of the one-day event that we have organized on a virtual platform. Then few other events as SPER online conference we have organized. That was also 2 days international conference. Professor YK Gupta sir was there, being a part and parcel of the society, and then he witnessed the event, blessed the event with his gracious presence. And recently, realizing the fact that at least with some situation we consider as better, but we have to take the corrective action. We have to be very conscious enough for the protocols for the COVID-19. We recently organized one event that was the physical event in Delhi Crown Plaza. That was on the occasion of World Pharmacist Day. So we have celebrated the World Pharmacist Day on 25th September 2021, and that was the physical event that we have organized after almost, I can say, 18 months. So I personally was very much excited to be a part of that particular event, at least to be with the young buddies, to be with my seniors, gurus, and all. At least that event has given me a hope that yes, maybe in the near future the things would be normalized. And I still remember the words that has been just said by Honorable Dr. YK Gupta sir as well. That event has to be physically organized. So indeed, sir, we very much interested to have this event and to have you on board in Mehsana. But unfortunately, we have to see different other sort of challenges as well, because we are not sure that whether the situation would be better or not. But at least we intended to organize this event initially as a physical mode, but then we realized the fact and then converted into the hybrid mode. But still, we are fortunate enough that very people of eminence like you, Dr. GN Singh, Dr. Mishra Saab, Dr. Dinesh Jaiswal Saab, and all Saranjit Singh sir, all have always been so nice for the society, that I still remember, sir, Dr. YK Gupta sir, the event of SPER, where in the first session also you have given the keynote address, and you continued to be in the event until evening, the valedictory function you have been there, and that was the Sunday, I guess. And I still remember the words that you mentioned, how you have managed the people to associate in the event for an entire day on Sunday. So that was something always given a hope that if we are able to spread our message, we are able to put forth some value addition in the existing science, existing research, definitely the society is doing something. And I, on every forum, every platform, I mentioned that it's not just organizing an event would be fruitful until and unless it won't have any such outcome. So definitely we are going to get all the minutes of meeting and all the report of this conference, so that whenever after completion of this 2 days mega event of SPER in collaboration with Ganpat University, we have to realize what we have achieved. On the next day, we have to start working for the SPER 11th conference, so that value addition has to be there in terms of academic, in terms of research, in terms of industry, and above all in terms of the society endeavors. Because no country will be able to grow until unless having hands on hand and then growing the society. No one individually can grow the society, but as a cumulative, as a consolidated, we can grow the society. And maybe I have certain positive, certain negative, but I'm least bothered about my negative. I have to endorse my positivity so that the positivity I do have, the benefits, the positive sense, I'll be able to contribute for the society. Definitely, I have to contribute. Definitely, we have to make it in that manner. I still remember the event because this is the decennial conference. So might be I am on very emotional note. I'm openly accepting this phase that this is a sort of an emotional moment for us. And I still remember the fond memories of SPER second annual conference that was held in Jamia Hamdard and in the month of March. And we were very fortunate that in the first day, that was the one-day event, and in the morning session, Mrs. Arshna Mudgal, then PCI secretary, was the chief guest, and she inaugurated the event. And on the same day, same event, in the evening, we have witnessed the presence of the legendary leader, Dr. G.N. Singh sir, along with the current DCGI, Dr. V.G. Somani and Dr. A. Ramakrishnan. So I still remember that we requested Dr. G.N. Singh sir that, sir, we request your presence to be a part and parcel of the conference. And he was so happy and given his consent. And we are fortunate to have Dr. V.G. Somani and Dr. A. Ramakrishnan as well in the event. So that way, it showed a commitment of these leaders that they haven't became a leader in a day or so. It has been a decade time to become a leader, to showcase the talent, to change the community at large. And we are on the same path, sir. We are on the same track. We are seeing you doing wonders for the community, for the Indian healthcare community at large. And you people have made certain changes in the community. People knows you very well that you have done a lot. And we are always very fortunate to have you on board for the event of SPER. And we are on our mission that how we will be able to add value in the community, in the society at large. The day one, I still remember when we established the research journal. So there was, I won't say that everything was so easy, because this is the 10th conference. I have to open up what it has been in the past 10 years. It was not easy. People said, there are journals, what you are going to do? There are many journals. What we are going to do having one new journal? We simply replied, we don't want to be one or the other journal. We just wanted to enhance the research, improve the research, and able to publish some quality research in the journal. And then efforts, they speaks. We are in PubMed, one of the greatest indexing agencies. Those researchers, we consider PubMed as one of the greatest indexing agencies. And from the volume first, issue first, we are included in PubMed. We are in PubMed. The current issue that we have already been published, that is also been included in PubMed. We are in Web of Science, the ESCI indexing as well. From the day one, we are in Scopus. Being an editor, might be I am more inclined towards understanding the indexing via PubMed rather than Scopus. But yes, we are in Scopus. So that reflected the commitment. We are very young association. 10 years was nothing. A long way we have to go. But somehow, someday, someone has to take a start. And at SPER, in the year 2010, we have taken that step that, yes, we wanted to, we have taken that responsibility to make the changes in the society and community at large. And nothing, no efforts would be wasted if wholeheartedly, genuinely, we are working in the profession. It's not easy. I wanted to tell this thing to the entire audience. It's not easy to have such an eminent people on board. They have lot many things to do. They have prior commitments as well. If they are here, then we consider this as our appreciation. We're not waiting someone to appreciate us. The presence of such luminaries, people like Dr. G.N. Singh, Y.K. Gupta, Sanjit Singh, B. Mishra, N.K. Ganguly Saab, they have given us a path, and we are working on the same. Women Forum has organized certain events, certain awareness camp for the breast cancer awareness, mindfulness. So these events are the need of the hour. It's not always that we have to talk about science every time we have to talk about research. The society and community are beyond the science and research. Whether we will be able to add value in the existing community, that is most important. Science has a lot to do, research has a lot to do. But as an individual, how we are going to contribute, what we have contributed, that is most important. If we are contributing wholeheartedly, then science and research will ultimately integrate. Considering the theme of the event, integrating academia and industry, we are very fortunate every time that we are able to get the support from Confederation of SIPI as well, CII, PhD Chamber of Commerce, Systopic Laboratories, Zuventus and all. They have supported like anything for one or the other events. They mentioned you are doing good for the community. Now we are in process of integrating some of the industry ready courses for the young brigade, so that these courses should be able to delivered by the industry people, and that information that is required by the industry, or that talent that need to be required by the industry, at least our BPharm institution or the master's institution, they will be able to get that info. Those students are able to get that information. We every time struggle and suffer that industry is talking about, we are not getting industry ready candidates. Institutions are busy in saying industry are not giving the opportunities. I guess both industry and academic institution, they need to collaborate. They need to have a great bonding, so that whatever the training they need to provide, they can seek the support from the industry. And industry at that time, they'll not say that we are not getting the industry ready candidates. If industry is willing to offer the internship, the support, the training to the institution, then ultimately they are going to groom the students in a better way. They are going to uplift the temperament of the students. And we as a responsible person from the society, we have to give them a hope that yes, there are industry, there are societies are serving for you, working for you, so that you can grow. And the moment youngsters will grow, definitely the nation will grow. No one person can grow the nation until unless we won't have a young brigade. So this is not just the responsibility of a society, of an institution or the industry. It's a collaborative effort that we have to do. And we cannot forget that it's not something that everyone is doing wonders for them and students. Ultimately, the person, the community to whom we are going to support, they should take the things casually. They have to be in that mindset, mindfulness that yes, they have to work, because everything we are planning to work for them. Otherwise, industry is working good. For what purpose they are going for the industry training or likewise? And institution, they are getting admission, they are offering research, they are offering degree. For what purpose they are engaged in this? But it is a human values that, and we in India, being an Indian, we have that kind of a mindset to support the society. So there are a lot many things that might be I do have in my mind, because it has been now 10 years. The society has given me that opportunity to serve as a national secretary from past 10 years. So it's always a proud moment for us, and it's a wonderful feeling every time to give the annual report of SPER in the annual event of SPER. So it's indeed a great pleasure to have you people on board. I extend wholehearted support, wholehearted thanks to all the organization and the organizer, Ganpat University, and the administration for making it happen. They have done a great job organizing this event in a virtual mode as well. So that showed their commitment. They are celebrating their silver jubilee, 25th year. So it's a deadly combination of SPER and Ganpat University to host a 10th conference and 25th conference. So that showed the collaboration of one or the other, and we have to work hands in hand. So with these words, I would like to one more time extend my heartiest thanks, and I would like to congratulate each one of us, because we have to feel proud. We are witnessing this event and we are a part of this event. So I would like to congratulate each one of us, including myself, to be a part of this event, to be a part of SPER, and look forward for your gracious presence in other events and your kind support to grow the community at large. Thank you so much. Jai Hind. Namaskar.
Congratulations to us, sir. Thank you so much, sir, for briefing everyone about what is SPER, what are the various activities SPER is taking throughout the year, and how it is beneficial to our students also. Thank you so much. We have a gracious presence of plenary speaker, Dr. Avni Amin, ma'am. We welcome you, ma'am. We rarely get this kind of opportunity to listen to the words of great knowledgeable person like Dr. Y.K. Gupta, President, All India Institute of Medical Society, AIIMS, Bhopal, India. I would like to invite sir for keynote address. Please, sir.
Namaskar to everybody here. And my sincere thanks to Dr. Pancholi and Dr. Upendra, and also to Ganpat University Vice Chancellor and their management for inviting me here. I will start my conversation. I will not say talk, but conversation with the remark which my friend Dr. Upendra, I would say dynamic Upendra, has made it, that this is perhaps a deadly combination of Mehsana or Gujarat Ganpat University, Mehsana, and the SPER. I would say this is a deadly combination of SPER and Ganpat University and Upendra, dynamic Upendra, and Dr. Pancholi, Dr. G.D. Gupta, and all those who are present here. This is really deadly combination. When I see so many galaxy gathering at a place which I would have preferred to be physically present, as he very rightly said, but I look forward always to visit Ganpat University if I can and witness the enthusiasm in person of the people here. I always say that we are producing the sufficient number of pharmacists, pharmacologists in India now. I would say to the extent that sometime if I see the number exceeds the demand, and that's why you are seeing that many College of Pharmacy, the seats are still very struggling, and who will get the full seats is not the criteria, but who will get the best students, and that will depend on the best reputation of the institution. And the best reputation of the institution is directly proportional to how the students of that institution are trained and how they are placed. And not only placed, what caliber they are enjoying. Now, this is directly proportional to what sometime Infosys Chairman Murthy said. We are not interested in how much quantity we are producing, but we are interested in how much quality people, which are employable people, you are producing. Today, employability means self-employment or employment in any institution. It will depend upon sorry, my apologies. Will depend on that how you do things differently than others. And I always quote this. In childhood, we were told one simple statement: leak, gaadi chale. You know, leak, when the bullock cart moves, it makes some sort of a pagdandi, or what you can say, path. Leak, gaadi chale. Leak chale kaput. Those who are not good boy, not kaput, you I think understand. Bin leak tino chale. Sire, sing, saput. That is what our today's pharmacy graduate should work upon. Innovative thinking, innovative working, and always constantly look at the innovation. And that is what the theme is. Innovation in healthcare. And unconventional reading, unconventional working, not syllabus. I always say syllabus in B Pharma, M Pharma is just to pass the examination, get the degree. But to be a successful in profession, to reach the ladder on top, you have to work out of syllabus, beyond syllabus. And beyond syllabus, you can only do when you will know what is the contemporary requirement of the country. And that is Your primary responsibility is whether you are fitting into the domain of requirement of industry. One, the requirement of regulator. You want to become a regulator. Domain of a requirement of a medical writer. Domain of a pharmacovigilance professional. Innovate in the epidemiology. And this is one of the leading pharmacists just now sitting in my office from Geneva, is a pharmacist. He is the top of pharmacovigilance man. The top pharmacy people are now heading so many industries, giving the institution, making the institution, giving jobs to hundreds. So instead of job seeker, you will become job provider. Why we say the early exposure of industry requirement, industry philosophy, knowing GLP, knowing GCP, knowing GMP at the early stage will make you a value-added person, will make you more required people, more sought-after people at industry. And I chaired a recently high-powered committee between how to improve industry, academia, innovation, which was made by Department of Pharmaceutical Sciences and has been submitted to Ministry, has been submitted to Prime Minister's office, and that has become a document now. In this, there are academic institutions, professors, NIPers, directors, and all stalwarts of industry, the top. And 3-4 recommendations I will just give you. A, that in each academic institution, pharmacy institution, there has to be incubation center, which should be voluntarily supported or inhibited by the industry. Industry should come forward, and industry will never come forward unless you make them comfortable at your campus. It's not that you approach the industry, the pharmacy institution approach industry only at the time of campus placement. Industry should be part and parcel of the curriculum development of yours, invite them as a lecturer, invite them as an examiner of your institution, and ask them to invite your students to take your students 5, 10, whatever they are, as an intern for 1 month, 2 months, 3 months, early maybe in M-Pharma first year, second year, and give them the project which interests them. Which interests them should also be the topic of M-Pharma thesis. At least 50% of M-Pharma thesis should not be the conventional me too, that my superiors, my senior has done this, so I am just doing that. Work, talk with industry and see that what problem they are working upon, see that what pharmaceutical innovation they are working upon, try to give them solution through these people, through their M-Pharma. They will get interested into industry. Industry is now looking for innovative things. API is one of the challenge. The whole industry was looking with academia support, how we can solve the dependency on API on China and other parts. Can we develop a cost-effective alternate method of development of API or the substances which are essential for creating these medicines? Can the academic institution, pharma institution, give us the alternative formulations which have easy penetrability into brain? Nano formulation, the NIPers are the bigger institutions and they are working well. Now their role we have casted is that each NIPer should handhold the middle or the smaller cities pharmacy institution. They should also have a mentorship role. And the mentorship role should also come from the academic institution, bigger academic institution, as well as the industry. Now, when the industry will know that you are potential, I'm not saying that you are fit, the academic institution is potential, then industry will also try to make you as a capacity building exercise. That is what the CSR funding we have proposed, that the industry will also put some sort of a money to capacity building of the academic institution. ICMR, DBT, DST, all are encouraging that academic institutions should work with industry, and such projects which have industry academia collaboration will get highest priority for funding. Because when we know that this is the project which a pharmacy institution is sending, has already endorsement for the industry, and industry will not invest money of even 1 lakh rupees unless it sees the worth in it. So best evaluation can be if it has industry funding, not only industry endorsement. So that is what is required. And now you see what the country needs today. The industry needs a person who knows regulatory process, a person who knows the good manufacturing process, the person who knows GCP, so that when an person goes, he is not on the floor and getting trained on a statistical method or on how to handle animal. These are the things which the academic institutions should make an effort, that you are exposed into this already. I think our institutions and need to slightly orient ourselves not to only provide theoretical knowledge, but also provide the process or the ignite, what somebody very rightly said, ignite the brain so that you think differently. You are good in communication, you are crisp in communication, and you can understand, analyze, and you can curate much faster the need of today's requirement. You can forecast the requirement. You can predict that after 10 years, after 5 years, which are the area where you need to work upon. Can you do the artificial intelligence to identify which antibiotic will be resistant and which antimicrobial agents will cause AMR, and you have to develop antibiotic in this sector. You, as an academic institution with industry support, we should identify through artificial intelligence or through mega search that this is how my new drug is going to be positioned as compared to available drug. Market forecasting, competitive intelligence, and the access, how will you increase the access? So these are the things which our Pharm D students should be able to tell us, should be able to do it. And these are the simple things which the industry academia interaction strengthening can do it. So now, to conclude, I would say, I compliment Dr. Pancholi, and I compliment this SPER society is doing such an excellent. And when I was listening to Dr. Upendra, the report, I will only say, if there are few more Upendra, if there are few Dr. Pancholi and GD Gupta, I think it will be much, much good for the pharmacy profession. This will definitely improve the status. This will definitely improve the caliber and of the pharmacy graduates, and this will improve the industry academia interaction, and we will create the workforce which will not only be the best in the world, but which will be, what you say, ready to serve on the plate. That means the human resource which will be produced will be ready to work at the place where they are deputed, be it industry, be it a regulator, be it a scientific writer, or be it a research laboratory. So I wish everybody a great success, and I will see that I am next time I am in Mehsana, and I also look forward for an opportunity to meet each and every of you, though I can meet through this couple of my good friends like Dr. Professor Ganguly, my guru, and Dr. G.N. Singh, and so many others. It's a wonderful opportunity for me. Though it was a virtual meeting, I enjoyed. Thank you very much, Dr. Upen. Thank you very much, Dr. Pancholi. And my apologies that I could not join early because there was a parallel meeting. So thank you very much. Namaskar.
We are looking forward for that time, sir. We will be together here physically. Thank you so much, sir, for a really inspiring speech. On behalf of our institute and SPER, we would like to present you a statement of gratitude. Thank you, sir. I would like to invite Professor Dr. Harvinder Popli, President, SPER Women Forum and Director, School of Pharmaceutical Sciences, Delhi, Pharmaceutical Science and Research University, New Delhi, to say a few words. Please, ma'am.
Thank you so much. And, first of all, good evening to everyone and all the dignitaries, starting from Dr. Mahendra Sharma, Dr. B. Mishra, Dr. Y.K. Gupta, Dr. Ganguly, Dr. G.N. Singh, Dr. N.R. Sheth, Dr. Jaiswal, and Dr. Upendra Nageche, and Dr. S.S. Pancholi. Dr. Nagendra has given a very, very emotional speech. And, since, I have seen him from so many years, I think almost 5 years now, where, where he's actually doing a lot of work in this industry. And it's a, very important topic today. Academia and industry, they are the two extremely important pillars of the economy, and a robust collaborations between the two augurs well for the country, enabling innovation as well as growth in the education system for producing an employment-ready workforce. Like today, we are talking about employment-ready and the skills. So in developed countries, the cutting-edge research often starts in the university, and the corporates frequently reach out to these academic institutions to co-create innovation solutions for addressing therapy gaps. But in India, this link has traditionally not been very robust, and the industry players have generally relied on their own internal research rather than reaching out and partnering with academia for solving their science and technology problems. So the social environment, which is stereotype image of pure science focus of educational institutes and the corporate emphasis on immediate problems, have given rise to the prejudice against the collaborations, leaving India much behind. We talk about China and Western developed nations. So, however, there is beginning a shift in this approach, which is very quite heartening. And some of the corporates and educational institutions, they are taken significant stride towards collaboration for innovations. And, in our university, which is the first pharmaceutical university, we had seen that, and I spending almost 15 years in industry and then coming into academia, I could really feel that, this change is now being kind of, happening. When we talk about the healthcare industry, it has been, a slow adopter of a technology, even though slow, but there is a steady adoption. This has led to the use of high end of technologies like AI, ML, neural network for the faster and the better diagnostics, data management, and develop the customized, healthcare solutions. If we talk about India as a startup culture, we had seen it is the third largest startup ecosystems in the world. And few years back, a startup, valued about $1 million or more, it was very rare. But, we have a thing like a unicorn. So over the past few years, we have seen that the list of Indian unicorn is increasing. So this is a hot harbor for startups. And with that, even the healthcare is making a very a good progress in this. And we had seen that, we are being, the India is being led, although, we are a little behind after the Germany, UK, US, South Korea, and Japan. But I think there is a vision that by 2025, we'll have 100 unicorns. So with that, it is very important that if we have innovations, and especially in the healthcare startups, we integrate the academia and industry. We had already seen that the nanotechnology is already leading to a dramatic improvements in healthcare. and scientists are using nanoparticles to target tumors in drug delivery systems and to improve the medical imaging. And some nanoparticle-based treatments are multifunctionals. They can find tumors and carry drugs for treatment. So academia, I can say, is a source of much potential radical innovations today. And it's often funded by grants from industry as well. And the partnerships, that has evolved between industry and academia will be leading to our innovations, which is reaching the marketplace. I think very well said by Dr. Y.K. Gupta that, our funding agencies are also seeing that, the industry and the academia collaborate and they are given a priority. So the converting the intellectual capital into an economic capital is a challenge for both industry and academia. And often the innovations, they have languished into a prototype stage without being driven to the further and to the value. So that is how, there is a lot of emphasis which is given by a government on a skill development and also the startup. So in countries like US, the corporates reach out to universities. So here we had seen that, they have started. So this shift is beginning. So although, there have been a huge leaps taken by various corporates and academias to bring about this positive change, the country is currently at a juncture where both industry and academia, they are playing a good role in a transformational story to reach the, new heights of innovations through the industry academia collaboration. So I am really congratulate to the entire team, to taken up this theme as a decade theme for the SPR. So there are extensive efforts to convert these innovations into a job creating commercial products, which is very essential. And, it clearly indicates that it only can happen with effective collaborations between industry, academia, and government, because funding is very important. So university innovation centers, incubators, there is a lot of funding which is given by the BIRAC. And we had seen that, in the 75th year of independence, it's recently been launched that incubations or startups, there is a lot of promotion or funding which has been given, by the funding agency. So academia has excelled in the incubations and research of innovative ideas, and in recognition that there is a more value in innovations than as an end product. So there are, many examples. So with the time is so short. So I just wanted to say that it's time we to ponder on the questions that on how can we have an innovations community, reinforce each other and leverage our strengths. So it's clearly a time to create or nurture an environment where the industry and academia can work hand in hand. And one of the questions which is posed by Dr. Upendra is that what we are doing as a society. So this is society act as a catalyst, to bridge that gap wherever we see there is a gap. So the society is there to nurture, even with a small seed fund, even with the small workshops, which makes, or even, the small competitions of students, which help them or which make them aware about that, how essential is that innovations and how can they get a funding, how can they do a research, how can they document research, how can they write what is the role of regulatory, what is the role of IP. So these all are the the catalyst or the small steps which brings them closer or which helps to bridge this gap. So with these words, I would congratulate the society for completing their 10 years, and also to the Ganpat University for their the Silver Jubilee celebrations, and hoping for the very fruitful and meaningful conference for entire students. Thank you so much.
Thank you, ma'am, for sharing your knowledge with us. On behalf of our institute and SPER, we would like to present you a statement of gratitude.
Thank you so much.
Thank you, ma'am. Moving ahead, I would like to request all the excellences on the dais to release the convention souvenir. Please, sir. Marshall Proust once said, "Let us be grateful to people who make us happy. They are charming gardeners who make our souls blossom." On his words, I would like to invite Professor Dr. S.S. Pancholi, sir, Executive Dean, Faculty of Pharmacy and Faculty of Science, Ganpat University, and Principal and Professor, Sri S.K. Patel College of Pharmaceutical Education and Research, Ganpat University, to propose vote of thanks. I also request sir to brief everyone about declaration of MOU signing intent with ISF College of Pharmacy, Moga. Please, sir.
A very good afternoon to all present here in auditorium, the dignitaries on the dais, the dignitaries, the luminaries of pharma field present online. It is really a privilege to extend vote of thanks to this elite dais who has illuminated the profession through service throughout their life. This is a very pleasing moment. I would like to announce that when we say the collaborations, interactions, and joint ventures are the need of time, when we say the theme of SPER 10th Annual International Conference and Exhibition being organized here in Ganpat University offline and throughout the internet world virtually in collaboration with SPER. The theme is we need to innovate in healthcare. We need to interact with industries. And a pride moment for us is here that two very prominent institutes, one, the ISF College of Moga, Punjab, and another, the Sri S.K. Patel College of Pharmacy, Ganpat University, both have agreed to sign an MOU to share their facilities, to share their expertise, to interact, to exchange the students, to exchange professors, and to exchange the knowledge. So I would like every one of the present people present online and offline here to cherish this moment, that this is a big agreement. One of the Prime Institute of Punjab, it is ranked 29th and NIRF ranking of 2021, and one of the best colleges of Gujarat. We are shaking hands together to serve the profession with more vigor. So yeah, Dr. G.D. Gupta is there online. I would like to congratulate, and I would like all dignitaries to applaud this event.
So thank you very much, Dr. Pancholi ji, and I am highly thankful to SPR. So both we are here, Dr. Narang ji and myself. So thank you very much, and I hope that this MOU will be do some of the new innovations, and no doubt the both institute, one of them is the in Gujarat and one is the Punjab. So we will work together, as we know that at the initial level, I have visited the Ganpat Institute. This is the S.K. Patel College of Pharmacy in 97. And I did work with the Professor S.S. Pancholi ji. So we again work together, and this MOU will do some of the new things and new innovation, entrepreneurship, as well as we will do in the good in the research also. So thank you very much. And I am thankful to the SPR and thankful to the all organizing committee. Thank you very much, Dr. And congratulations to all of you.
Moving to the this privileged part, I am here to put vote of thanks to all the dignitaries. First of all, Honorable Shri Ganpatai Patel, President of Ganpat University and Patron-in-Chief, whom we lovingly called Dada. Though sitting in California, Dr. Ganpatbhai Patel, Padma Shri, he has been a source of energy, source of motivation to all of us. And he was very happy to learn about this conference. He congratulated us. He passed a message to be read that we are doing great by convening such a conference where such luminaries would be coming together to basically guide the new generation. See, during this conference, we are making an effort to bring together the generation which is above ours. Actually, I would say we are middle generation and the new generation. And there is a pool of knowledge with seniors, as you have seen Professor N.K. Ganguly, Padma Bhushan available today to talk with you, which is one of the finest moments of my life. I could have heard listening him live, but of course, because of this pandemic, we were to attend this conference virtually. But really, I am very thankful to Honorable Dr. N.K. Ganguly sir, Director General ICMR, for having spared time and being present here to guide all of us. Not only, I would say, Professor G.N. Singh said he is his guru. So he is like our Dada Guru who came here to bless us. So Ganpat Dada and our Dada Guru, Dr. N.K. Ganguly, actually we feel privileged to have your words of blessing. Professor Dr. Mishra, National President of SPER, Dr. Harvinder Popli ma'am, President of SPER Women Forum, Professor Dr. G.N. Singh sir, former DCGI, Secretary cum Scientific Director of Indian Pharmacopoeia Commission, Dr. Bhashwat Chakrabarti sir, Chairman Scientific Committee of this conference, Dr. D.K. Jaiswal sir, Pharmaceutical Consultant at USA, Dr. Upendra Nagaich ji, Young Dynamic Secretary of SPER, our Professor and Pro-Chancellor, Dr. Mahendra Sharma sir, who blessed that during the welcome address in the beginning, and Executive Deans, Head of the Institutes, HODs of Ganpati University. It is really a pleasure to have all of you here on the SPER 10th Annual International Conference and Exhibition being organized by Ganpati University in collaboration with SPER, which is supported by Gujarat Council of Science and Technology, which cost, it is a financial support they have provided. The innovation in healthcare, integrating academia and industry, the theme of this conference brings forth the need of the day, need of the hour. And I am actually privileged to report here that Ganpati University has been proactive in collaborating with industries. We have industries in-house, and at the same time we have MOUs. As a pharmacy college, we have MOUs with more than 24 industries. Most of our MPharm and BPharm students, they are doing internships and they are doing research projects in collaboration with industries. Recently, we had signed an MOU with Gujarat Chemical Association and Gujarat Pharmaceutical Association, which has more than 700 member industries. And they have opened up the forum where we can interact, we can train their manpower, we can get our students trained there in the premise of their industries, and we can take up the problems which the industry is facing. And they, being an MSME industry, they cannot afford to do the research. Our students are fortunate enough that they will work on those problems, and we are confident that our professors will be able to deliver their commitments. It is really a matter of pride that we have been blessed by the specialists, eminent specialists of our field. Dr. G.N. Singh, he has been a great regulator who has made India feel proud by making our regulatory system more standard and more acceptable worldwide. We have with us online available Professor Dr. Saranjit Singh ji. He is better known as Stability Singh. He is for his contribution in the field of stability. As Dr. Singh, G.N. Singh mentioned, that contributions of Dr. Saranjit Singh. I wish you a very happy birthday, Dr. Singh. Today it's your birthday, I know. And
thank you. Thank you.
Yeah.
Thank you.
And Dr. Singh, we feel blessed. Actually, you have visited this campus earlier. I was there at that time also. And we are really blessed that you have agreed to be one of our plenary speakers. We have with us Dr. N.R. Sheth, Vice Chancellor of Gujarat Technological University. Sir has rightly pointed out that innovations are need not be incremental. They should, even our Vice Chancellor, Professor Mahendra Sharma, he expressed that we have to do breakthrough discoveries, and we at Ganpati University provide an ecosystem where not only we provide generation of creative ideas. Recently, we had a pharma ideathon where students were asked to submit the ideas that can be converted into prototypes. And Ganpati University's student startup policy, they had announced that first 5 best ideas would be supported with a seed money of rupees 2 lakhs. So that kind of support we are giving to the new ideas, and we are finding that such ideas are taken up by industries for commercialization. So this is the way Ganpati University moves, and this conference is a kind of step where we plan to move ahead in such a manner that the new generation of scientists in the pharmaceutical domain, they get acquainted about the recent trends in research, the recent needs of the pharmaceutical society and pharmaceutical industry, and they also come to know about various regulatory requirements. We have identified, and we are fortunate that renowned speakers have agreed to speak in plenary sessions, like Professor Dr. Avni F. Amin from Apotex, Canada, and the chair for that session would be Dr. Indrajit Singhvi, Vice Chancellor, Sai Tirupati University, Udaipur. Dr. Heng Von Sia Paul from National University of Singapore, and Dr. S.K. Jain from H.S. Gaur University would be chair of that session. Professor Dr. Saranjit Singh from Naipur Mohali. That the name is always added, Dr. Savi would be remembered with Naipur only. And Dr. Anuradha Gajjar from LM College of Pharmacy, Ahmedabad, she would be chairing the session. We have Dr. Vandana Patrawale's talk in a session. She belongs to Institute of Chemical Technology, Mumbai. And that session would be chaired by Dr. Dhiren Shah. Dr. Christian Grenan Ferre from University of Barcelona is to speak in one session. That is to be chaired by Dr. Manjunath Ghate from Nirma University. And we would be having a talk of Mr. Jigar Patel from Brillare Science, Ahmedabad. He is a young entrepreneur having established a world-class cosmetic industry that plans to compete with Nivea and others. And they have very good ultra, I would say, most recent technologies they are using into their products, and they have a very ambitious plan. His session would be chaired by Dr. Ali Asghar Sahiwala from Dubai College of Pharmacy, Dubai. So with these preliminaries lined up, to share their experiences. I am feeling very proud that not only my students, but more than 300 participants, the delegates, they are going to get benefited. They are from throughout the India. And at the same time, we have arranged for two scientific presentations, one in the form of oral presentations that are going to be held today in 3 tracks. And we have poster presentations to be done tomorrow from 9 to 10:30, again in 3 tracks. We have more than, I think about 59 exactly, the oral and poster presentations which are being accepted for presentations. And these young researchers, they are going to be sharing their researches. There would be comments from experts. The experts would be guiding them, and others who listen to these presentations will get an idea what kind of research is going on, how industry can take up this research, what are the needs of the time. We have, as rightly pointed out by the dignitaries, we have proven during the corona times that yes, Indian pharma, Indian talent is one of the best in the world, and we can produce even during the challenging times the good quality and innovative products. We have planned up these presentations to be evaluated by senior scientists and senior persons from profession of pharmacy. The award ceremony is planned tomorrow, again at the same time from 3 to 4 p.m. So till then, I wish that the enthusiastic delegates and researchers who have joined to seek and share the innovative ideas during this conference, they get benefited, they enjoy the learning, and it is the time to actually put on record the contributions that Gujarat Council of Science and Technology has supported this event by financial grant. We are extremely thankful to Guj Coast. We are extremely thankful to Society of Pharmaceutical Education and Research and its leadership and office bearers for having given us this opportunity to co-organize this event, which is a kind of world-class event. We feel we have tried to plan it in such a way that everyone is benefited, and we remember, we cherish this event for a long period of time. Thank you very much, and wish you all a very good learning experience.
Thank you, sir. On behalf of Ganpat University Faculty of Pharmacy, I would like to thank all the dignitaries on the stage and present virtually for their precious time and for sharing their experiences. At the end of this inaugural ceremony, I request all of you to please rise for the national anthem.
जन गण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड़ उत्कल बंगा विंध्य हिमाचल यमुना गंगा उज्जल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Jai. Thank you, everyone. Now onward, I would like to request Dr. Geeta Patel to hand over the session for next 2 plenary sessions. Please, ma'am. Geeta, ma'am.
Thank you, madam.
Ma'am, we'll have 5-minute break and then we'll start our first plenary session.
Sure, ma'am. Thank you. Hello, madam. How are you?
I'm good, thank you. How are you?
Fine, ma'am.
Am I properly audible?
Yeah, ma'am. Properly
Okay.
audible and properly visible.
Okay, perfect. Thank you so much.
Okay.
Dr. Avni ji, you have recognized to me. Good afternoon and welcome.
Yeah, good afternoon. Good morning for me.
Yeah. Good morning. So myself, Dr. G.D. Gupta, and I
Right.
was in LM Lachu College in Jodhpur. At that time, I worked with in your lab in with the Dr. M.C. Goel sir and Dr. G.K. Jani
Right.
ji, and you did
Yes.
lot of work related to this is the biostatics.
Correct. Yes.
Yeah.
Yeah, it was good working there, and now we are in the industry, but it's good to be back in the academia.
Yeah. So I am in ISF College of Pharmacy, Moga,
Okay.
in Punjab.
Okay, perfect. Good to see you, sir.
Welcome, madam.
So, madam, are you presenting your screen?
Yeah, I will. You want me to do it now?
Just for
checking, correct?
checking purpose.
Okay. And that's okay. So is that okay now?
No, ma'am, it's not shown on screen.
No.
Not visible.
Okay, 1 minute. Okay, let me check it again. Okay, window.
Hello, Paul. Thanks, sir.
Hello.
Sorry,
Can you
sir,
see
we are
me?
actually getting late.
Yeah. I have.
For inconvenience.
I hope we can finish in time because I didn't expect it to be this late.
Yeah, sir.
Can you hear me clearly?
Yes, madam, it's visible.
It's visible, correct?
Yeah, it's visible. Just
okay, perfect.
Shift it slide to slide. Mane just check.
Okay.
Slides or not? Yeah.
Yeah.
Sorry, madam. Fine.
Perfect, right? Okay, no problem.
Ma'am, due to time constraint and already the second speaker is already joined in the session. So we have a duration of the lectures for both the plenary session 30 minutes. Is it fine with you, ma'am?
Okay, I can rush through it.
Thank you, madam. Thank you for your cooperation and support.
Yeah, I think the slides will be more because they were made according to 1 hour.
Yeah. No problem,
But,
but it is my request to just highlight the important or probably 30 to 40 minutes. Try to finish it. Because
yeah,
after
I'll try for that.
every session, there are oral presentations and all.
Yeah, perfect. No problem.
Sorry for inconvenience, madam, please.
Yeah, that's okay. I understand. So I think if I share my screen, I cannot see the video. Is that correct?
Pardon, ma'am?
If I share the screen, I am unable to see the video. Is that correct?
No, no, no. You will be visible on the screen.
Okay. No, means I cannot see myself and others, right?
Because the moment you will be getting the files, the presentation on a presentation mode, so most probably the video might be hide if it is not in a gallery view.
If you are presenting on whole screen, then you might not be seen on screen.
Yeah, whatever. That's fine.
Okay.
Because we don't use the Zoom very much, so I'm not sure about all its features. Also, I was not sure, does it have a pointer or something? Like we can highlight something, is that possible?
Actually, in Zoom problem.
Yes, madam, you can select a pointer. On the left hand side, you can click in your mouse or computer. So you can select the highlighter, pointer, it is available. And in the Zoom meeting, both options, so you don't to go to the full of screen, then you are the visual full time.
Okay. 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 Thank you, sir, so much for that. Let me just try it.
Should we start, ma'am? Before starting, I'm introducing you to the attendees.
Yeah, you can see my screen, okay? You could see my screen, right?
Yeah, currently it's not.
No, I just now
stop the presentation. You currently stop the presentation, right?
Yeah, I did. Now I am okay.
Fine. After your introduction,
sure,
you can carry on.
Okay, no problem.
Okay, fine, ma'am. Welcome you all in first plenary session. We have Dr. Indrajit Singhvi sir as a session chair, but due to certain unavoidable circumstances, he has to leave the meeting. Dr. Indrajit Singhvi sir is currently President, Sai Tirupati University, Udaipur. Today we have with us Dr. Avni Madam. She is scientific leader, formulation development, Apotex, Toronto, Canada, as an eminent speaker, and he will deliver her expert talk on quality by design in pharmaceutical formulation. Madam has completed her PhD, Master and Bachelor degrees in pharmaceutical sciences. She has strong technical expertise in pharmaceutical formulation and manufacturing processes. She has expertise in pharmaceutical development for abbreviated new drug applications using quality by design. Possesses extensive experience with GMP documents, product reports, process investigation and stability testing reports, master records, standard operating procedures, work instructions for complex product and processes. She is proficient in writing investigation deviation reports with strong technical background, trained in investigations and change control as per GMP requirement. She is proficient in statistical softwares like JMP, Minitab, and utilize them to identify product issues and trends. Expert in technical and investigation report to ensure maintenance of product quality. She is specialist in CGMP, FDA, Health Canada, EMA, TGA, SUPEC, and ICH guideline. She has experienced regulatory audit program. She is also associated in teaching of theory and practical courses for Seneca students and for Apotex Seneca courses. She was former principal and IC director pharmaceutics at the Institute of Pharmacy, Nirma University, India from 2004 to 2010. As a part of her special achievement, 3 Indian patents has been granted and she has written various research publications in reputed international and national journals. She awarded 3 gold medals by Gujarat University, India in 1990. Granted GP Nair IDMA Award for the year 1990 for securing first position at Gujarat University, India. Honored the Motan Devi Dandiya Prize in Pharmacy in 1998 Best Publication Award. Thank you very much, ma'am, for your gracious presence, and I welcome you on behalf of my institute to deliver your first plenary talk on quality by design in pharmaceutical formulation. Please, ma'am.
Thank you, Geeta, very much for the kind in this thing. And at the onset, first of all, I would like to thank Dr. Rakesh and Dr. Pancholi for inviting me for this conference. I wish the conference best wishes. As I go about my talk today, I am going to give you a brief overview on the quality by design in pharmaceutical formulation and development. I am related to the generic industry and my topic will go from there. First of all, before I start my session, I'd just like to thank that all my learnings are indebted to my PhD guide, my guru, Professor Dr. MC Gohel, who will always be a source of constant inspiration to keep learning newer scientific concepts. So we really miss you, sir. I also would like to acknowledge Dr. Keyur Patel and Dr. Naveen Vaya from Apotex Inc., who have helped me in this presentation. And contents for the today. So we're going to quickly look at what is quality by design, understand its terminologies, also understand the risk assessment. And I have a case study to give you on the solid generics, how a quality by design can be applied in the formulation development. So let's take a look, first of all, what is quality by design? So quality by design, as you may be all using the word QBD.
Sorry to interrupt you, ma'am. Your PPTs are not visible on screen. Have you shared it? You present it?
I have it. Just let me do. Okay, let me just go back. Okay. How about now?
Yeah, madam, fine. It's there.
Okay, is it okay now?
Yeah, madam, fine.
Okay. Okay, sorry for that. So you may have all heard about QBD, quality by design. Basically, what is quality by design? So it is a systematic approach to developing any pharmaceutical product. And in this quality by design, we are going with predefined objectives. And also the way the quality by design is made is that we want to emphasize on the knowledge that we have from the understanding of the formulation and the process. And overall, the quality by design is based on sound science and risk management techniques. So in that case, let's take a look. Let's take a look at, what is there in the benefits of the quality by design. So when we apply quality by design during the pharmaceutical formulation, during the pharmaceutical development, we will find that your overall quality of the product you will always have a better quality of product than the one which you did without the QBD. And also, as I mentioned, it's based on science, it's based on information, and it has a sound foundation that will help the product not only during the development stage, but also as you go throughout the life cycle of the product. And when we have quality by design, okay, I'm having some difficulties in moving my slides. Just give me a moment. So when we have, when we use the quality by design, as I mentioned, you will have improved quality. And from the regulatory aspect of your submission, you will find that there will be overall fewer cycles, faster review and approval process, and fewer supplements to file after you have got approval. And as I mentioned, during the life cycle, there will be fewer batch failures, less problems in stability or problems of withdrawal. And taking a look at what the essence of quality by design is based on. So quality by design is based on these ICH guidelines, which are then taken up by FDA, Health Canada, EMA, and these guidelines, the ICH Q8 R2, the Q9 and the Q10, they give us all information about this quality by design. And we are going to take the concepts from this ICH Q8, Q9 and Q10 and apply them to our development in such a way that we get a consistent and a good quality product. So what are we doing when we are applying this quality by design? So you will see a lot of terminologies when we are using here, because those are the wordings that we are using in and out. And what we do is initially we start with identification of the quality target product profile. And then we have to define the critical quality attributes of our product. Then we perform risk assessment as given by the ICH Q9. And then we identify the CMA, that is the material attributes and the process parameters. And finally, we develop a control strategy which is utilized for the product life cycle. So with this introduction about the QBD, we will take a look at what are the basic definitions, and we will understand what we need to define in each of these particular aspects. So first of all, as I mentioned, the quality target product profile. So what is this? Basically, this is a high-level prospective summary of the characteristics of the drug product. So whatever we are intending, a final product that we are intending, and this is the first moment that we want to pen down what is all the information we are looking for. So things like what type of dosage form we want to make, whether we want to make a capsule, whether we want to make a tablet, whether we want to make a solution or a parenteral. And then we want to find out that what are its attributes and how are we going to prove the bioavailability, what type of clinical studies or what type of bioequivalent studies will be required, because ultimately our development is going to be based on all these criteria that we have initially defined. So that's what is the first thing, and we will take a little bit more look how we can define this quality target product profile also. Then we have the very important attribute that we define, that is your critical quality attribute. So what is critical quality attribute? It is basically the property of the drug product that we are looking at a holistic approach from physical, chemical, biological or microbiological property. And these are the ones which are going to be impacted by the way we use ingredients in our formulation and the manufacturing process that we apply. So this is going to be very critical for us, and all our development is based on getting a proper critical quality attribute. And in this, the things like assay, dissolution, content uniformity, all those type are going to be utilized. Then we have the critical material attributes. So whatever drug substance, formulation, in-process materials we are planning to use, we have to understand their property too. because their property is ultimately going to have an impact on the critical quality attribute of the finished product. So we will take into consideration the drug substance, the excipients, the in-process materials property, and we will link it with the critical quality attribute of the drug product. So as you see that now we are going with a holistic approach with all the information upfront, and that's why it is going to ensure that we are going to get a quality product. And the same thing is going to apply for our manufacturing process. So when we define our manufacturing process, we are also going to consider which are the critical parameters which are ultimately going to have an impact on our critical quality attribute. And we know that these are the critical parameters, so we will be able to really control them and ensure that they are running properly. So ultimately, as we see, it is a combination of this critical material attribute of your raw materials, including drug substance excipients, all your process parameters, which you are going to think about in terms of the critical quality attribute. Again, these critical quality attributes which have an impact on the quality target product profile. And moreover, as I have written over here, it's very true that it is better to be quality by design than rather than do quality by testing. We don't want to end up in surprises when we do the final testing of the product that it's not correct or it has some failures or it has some OOTs. So if we incorporate this QBD in our formulation development, we are definitely going to see a good quality product. And then as we defined our concept of conducting risk assessment. So what is this risk assessment? Risk assessment is a systematic way of identifying the risk, because now we are aware of these concepts. You are aware of the concepts of CMA, CPP having on CQA. So you have to identify the risk, that what is the risk of these parameters on the critical quality attribute. You analyze the risk, you evaluate the risk. So all our studies that we are conducting, the optimization that we are conducting, the formulation development we are conducting is going to be based on this risk, and we are going to use the high risk or the medium risk which are going to be beneficial and it is going to be useful for us. And when we see in these guidelines, there are various techniques which are utilized for doing the risk management. So mainly over here, as you see, you could use any or combination of these techniques which are here, like the failure mode effect analysis or the failure mode fault tree analysis, and all these in combination you can use or you can use alone. But mainly, as you know, that FDA has also given out the examples of doing quality by design development work for an immediate release product and a modified release product. If you are interested in QBD, those are like the Bibles of the QBD, which will give you all the information that you need and how you can approach. Even if you read those examples, it's easily available on the website. Those are actually using the risk assessment based on low, medium, and high. So they are ranking the risk relatively. So here we are doing a relative risk ranking, and we use this color coding of low, medium, and high. So we take all our parameters, all our processes, and we correlate them with low, medium, or high. And this becomes very easy for understanding, and also it gives you a visual look, and you can do your development work by just knowing which are the medium or the high risk parameters. Again, this will be on sound scientific knowledge that how we can carry out this risk assessment. So we will quickly go through one of the examples that we utilize for the generic industry, and we will see how we can define these material attributes, process parameters, and optimization. And finally, we can achieve a control strategy which is necessary as per the quality by the design. So as I mentioned, we're going to take a quick look at one of the solid oral generics. And in this, as you see that any generic development, if you take a generic formulation and a generic product, it is based on whatever is available in the market. So you usually pick up your brand, or you pick up the RLD, what is available, and from that you are going to define all these parameters, and you are going to charge your formulation and your manufacturing process according to whatever is made by the brand or the innovator. So in the first of all, when we do quality by design, there are two important things that we need to do. One is to identify what is the innovator product and to have a holistic look at the innovator product. And the second one would be to do the risk assessment and identify the QTPP and the CQA. So in this particular format, as you see, you do detailed investigation of the innovator product. And when you do detailed investigation of the innovator product, you take all the information from that, because this basic information that you have from the innovator product is going to help you in actually identifying your QTPP and CQA, because as a generic company, there are many factors which you are going to rely on the innovators. So we have to take all the information. So probably you can use the Orange Book to view the patents, you can use the label, you can use the FDA website, and there is a lot of information available to get information about the innovator product. So we start from there. We take a look at the innovator label or product monographs, whatever is there in the market. And we also have to take a look at all its packaging configurations, at how it is stored. And from that, we will get an idea if there is any degradation, if there are any stability challenges, or it requires some special packaging or some special testing. So from that, you will get an idea. And then we do the physico-chemical analysis. We we break down the product, and each and every parameter of the product can be viewed over here. So first of all, as we talked about, we are going to identify a quality target product profile. So what are in the quality target profile that we need to identify? So we identify things like the dosage form design, route of administration, dosage strength, and all its product attributes. And then, as I mentioned, stability, bioavailability, bioequivalence. How are you going to administer it? Does it require any sprinkling? Does it require any sprinkling on applesauce? So when you have an information all at front, you know that you have to match all this. So whatever product you are making, you have to make sure that all these attributes are going to be matched by the product you are making. So it's a good idea to write it down, to have a summary, and for that which you can do the backing. So in the second part of identifying the critical quality attributes, so these are the attributes which usually end up in your certificate of analysis. So they are usually ending up in your specification, and there are different guidances and different criteria under each. So upfront, when you are going to note down all your CQAs, that is going to be very helpful for you to actually take a chart that whether you need splittability study, whether you need alcohol dose jumping study, whether you are going to have any problem in degradation. So that is why this is the points that will need to be identified. And when you develop your CQA, things like physical attributes, identification, assay, content uniformity, degradation product, residual solvents, drug release, water content, and microbial limits. So these are the major items which go into the certificate of analysis and specifications of any product. So what are the steps that we will be taking in this development? So after identification of QTPP and CQA, you are going to now go in your formulation and process optimization stage. So in the formulation stage will be the identification of your material attributes, and for the manufacturing process is going to be your CPP. So you do the initial risk assessment as we talked about. You do the ranking, relative ranking of the risk assessment. Then you make a plan that which ones are of high risk. So you are going to do optimization for that. So you will optimize your formulation, you will optimize your manufacturing process, both from the concept of using any design of experiments, which we will little bit talk about later. Then you carry out the experiment, you find the best result with your experiment, you analyze it, and ultimately you are going to update your risk assessment that initially you started off with a high risk, but because of your experimentation, now you are able to reach to a lower risk. So we'll just quickly take a look at how this is going to be done. So this is just a placebo example of any modified release matrix formulation, which could contain a modified release polymer like HPMC K100M, a diluent like lactose, a binder like PVP, and a lubricant. So it's just a simple formulation just to give you an idea that which parameters and which material attributes are going to be important for us. And as a high level, we are going to use the dry granulation manufacturing process, and we are going to make a tablet out of it. So these are the corresponding steps that we need to utilize while doing our manufacturer. So each step has to have a control, and that is why we will see the importance of this applying this QBD under each step. So first of all, when we take our formulation, we have drug substance, we have excipients, and we will have intermediate products as we do the manufacturing. So in the drug substance, we have to identify, as we saw, all the physicochemical properties of the drug substance, and we have to link them that are they going to impact on the CQA or not, and which ones are impacting, then we will do detailed study of those. Then you have excipients, and in same way, you take the properties of the excipients and you link them with the CQA. And then if you have any intermediate product like a final blend or a powder blend going into compression, all those parameters of those intermediates are also going to be important, which need to be controlled. So this is a very overview example of how you can do a drug substance risk assessment. So as I mentioned, each drug substance critical material attribute like particle size, flow, solubility, hygroscopicity, etc., they are taken into account. And then if you see to your right column, you have your assay, content, degradation, dissolution. So each parameter of the material attribute of the drug substance that you have, you are linking them with the critical quality attribute, and you are identifying whether it is low, medium, or high. So this is the initial risk assessment that is being conducted on experience, sound knowledge, scientific understanding, and based on the knowledge. And now you know that there are certain things which are medium and high. So that means your formulation optimization requires these studies to be done, and you will focus yourself on these important parameter of study that you will be doing. Similarly, for the formulation also, that what level, what grade of the formulation, like the grade of the modified release polymer, as in this we have used HPMC K100M, whether we can use 4M or whether we can use other polymer, that we have to assess. And usually we know that these polymer, modified release polymer grade level, they will have an high impact on the dissolution and the tablet splittability. So this type of assessment for the formulation composition you will do, and now you know that which are high, which are medium, and according to that you can then think of doing these studies. So you will study the level of the polymer, the grade of the polymer, level of diluent, binder, lubricant. So all this will be done. Then you have the excipient properties. Again, modified release polymer over here. So very important. We have to take a look at its physico-chemical property and how it is going to impact our CQA. So similar type of risk assessment is also done for the material attributes of our raw materials. So you have your modified release polymer particle size, viscosity, and you have the diluent particle size, binder particle size, lubricant particle size, and you will do an assessment of this on your CQAs. You will identify them as high, low, medium, and accordingly you will go forward. Similarly, you are going to use this concept even for your manufacturing process. So this is, as I mentioned, the high level of the manufacturing process. You have processes like screening, blending, dry granulation, lubrication, compression. And for each intermediate step, what you need to do is you need to identify your process parameters. So some examples of the important process parameters for each intermediate step, they are aligned with this. So you can take a look that if you have blending, the blender type, the blender speed, the blender time, all is going to be important to ensure that you have a good quality product. So for each intermediate step is the step of identification of the process parameters, and then you will be doing the similar type of the risk assessment. And based on this risk assessment, what you can do is you can plan your optimization studies, that you will need to do a blending study or a blend time analysis or a blend speed analysis. You will need to do a design of experiments on the compression, on the dry granulation. So now you know what is important, and that is how you will plan your design of experiments. So when we are doing these optimization studies, there is a utilization of the statistical tools, which are holistically going to give you a lot of information. So when we are using these statistical tools, we are using these control charts, we are using this design of experiments, Pareto charts, and also the use of the process capability analysis. So this all is going to give you that when you are applying your design of experiments from all the batches that you have made, you can do systematic statistical analysis and come up with proper parameters which are going to be best for the product, and this will all give you information. So when we are using this design of experiments and when we are planning out, we do and we choose them in such a way that we are able to do like multivariate approach, because as you have seen that there are so many factors, drug substance factor, drug product factors, it is good to apply a multivariate approach in doing your regression, in doing your analysis, and that is going to bring you out that the proper input materials that you have is going to give you an output, is going to give you a response. So we utilize this knowledge, we utilize stats, we utilize our information, and we find out that how this multivariate approach is going to impact our product, and which are the interaction terms and what are the important terms which are going to be risk based. And we will identify these factors which are going to be critical, and we have to put controls onto these factors. And you can use variety of designs, although I will say that in research it is possible to use these big designs, but when you are in a generic industry, usually you are short of time, you are short of resources because you want to reach the market very fast. So sometimes it becomes challenging to use these big designs because our API is very expensive and we may not have a lot of opportunity to use these big designs. However, simple designs are also very useful, which give us a lot of information, and as per the need, we can match that which of these designs we can accommodate. And then we will utilize these statistical softwares like Design Expert, Jump, Minitab. There are many softwares now in the market which can be easily used for this purpose. And these softwares are also able to auto generate these plots, And these plots give us like a pictorial view that from that easily we can see that which are our critical factors, which are the ones which are going to be impacted, and we take a note of that and we are going to put a control on them. So these counter plots, response surface plots are very much recommended to be utilized when we are going to do the study. When we plan the study, we have to show to the agencies that how our equations are behaving and which are critical, and we have to give them confidence that we are going to control them. So we are going to need to put acceptance criteria based on our analysis. We may need to put acceptance criteria towards our drug substance, excipients, our in-process materials, finished product, and finally we have to achieve our CQA and the QTPP. And this will give us an opportunity to identify the design space that we are looking for, that the ranges, for example, of the compaction force or the compression force that we are looking for. Now we have sound knowledge and we have experiments by which we have proved that this design space is going to be good enough. So when we are going to update our risk assessment, you can see over here that many on the basis of many of the studies that we did, these red colors, actually they become green color, but I kept them red for you to take a quick look. But I have put in that, see, the blender speed is fixed, the time is fixed, the non-critical within design space. So I have put this, and the agency would like to see this type of assessment completion after you do the initial. So this is a type of your updated risk assessment which is going to be based on your initial risk assessment. And then finally, you come up with a control strategy, and the control strategy should be going throughout the life cycle of the product. So you will identify all the controls that you have for your input materials, for your drug substance, for your excipients, all the controls on the manufacturing process that you are going to put. You will list them down in the control strategy. You will give them the ranges. You have to show them that how you optimize those ranges, where did you bring them from, and finally you have to show them what are the controls on the finished product also. And this control strategy is usually presented in a tabular format in the pharmaceutical development report. And as I mentioned, it can change if you go for technical transfer, if you go for validation, the stage 1A, 1B, 2. At all points, the control strategy needs to be live document, it needs to be updated, and it needs to show the latest information that can be obtained through the series of experimentation that will be performed. So this is just a example how you can have a table as shown by the FDA examples that you can have in your control strategy. And then finally, we come to the summary that, as you saw over here, QBD is the need of the generic pharma industry. It is a requirement by regulators, and also it's not only based on new information, but on sound knowledge, on science, on experience. And overall, when we are applying this QBD tools, you are ensuring a good quality product, which is going to have a faster approval cycle for your product and a fewer problems during the technology transfer and the life cycle of the product. So at this, I thank you again for giving me this opportunity to share my views. And I hope I rushed through the slides properly and I am in time for that. So thank you very much. It's open for discussion.
Thank you very much, ma'am, for covering all aspects of QBD in such a short time and explaining role of QBD in pharmaceutical formulation development. Now session is open for discussion. Attendees can put their questions in question answer section or in chat box.
Thank you, madam. Your session was very informative, and I hope all the delegates must have been benefited out of it. we are really sorry to cut short of your allocated time. Actually, it went, the inaugural ceremony went long, and the next speaker was ready, and he has to go for another meeting. So we had to cut short for that. I am really sorry for that.
That's okay, sir, as long as I covered everything. And I hope the students are going to benefit from this, because we are now not into a lot of research, but this is the need of the generic industry. And it is what actually we are doing at our hands. So it's a good opportunity to share to the young students that they can, put their time, say, mind frame according to this, and they can pick it up and utilize it in their life.
Agreed, madam. And that is the basic thing which is required, whether it is generic research or any formulation you are making. You need to know the experimentation design and the pre-formulation parts that remain all together same for everything. So thank you very much, ma'am.
So I think we have a question here. Somebody is asking, Madam, is QBD is mandatory for any product approval regulatory bodies? So I will say that it's not mandatory yet, but if you see the way the questions are being asked by the regulatory agencies, you will find that it is going to be like, it is requirement of the day. So if you don't do, then there will be a lot of questions, and they will ask you that how did you arrive at these parameters, or how did you come up with these values. So it's not a mandate yet, but I will say it's very near to mandate, because it is a part of the guidelines. It is a part of the ICH Q8, Q9, QT. So definitely the agency is expecting, and I will not say that it's that they will allow your submissions to go without doing all this. So they will definitely ask you to do this type of assessment for your formulation and manufacturing process. So I will say, yeah, very near to mandate.
But madam, say for example, someone has not done the QBD and they have come up with a good product. So is it like that
good product,
they will not get the approval?
Yeah, so good product, Rakesh sir, it's a holistic approach, right? When do you say a good product? So from the regulator angle, a good product is only when it is going to have all the information aligned to it, right? If you cannot prove that your product is going to be okay within the like levels of the modified release polymer, if you cannot prove that, if you in your big batch you are not able to get the correct amount of the modified release polymer, if you are within the SUPAC guidelines, what is going to happen? So if you cannot prove all that, then the regulators will not be happy, and how will they consider your product to be a good product? So a good product is not only when you have like a comparative bioavailability study. It's not that that's a good product, that you have a good bioequivalence. That's not the end of the product. All these have to go inside the product. You have to have these controls, and you have to come up, how did you come up with them? So they want to see all that, and they want to be satisfied, because they will ask all these questions finally, because they want to see that in the life cycle, when you are doing large scale and commercial production, they want to see that your product is robust, and you are going to make the same type of the batch. That is a good product for them.
Yes. Thank you very much, ma'am.
Yeah, you're welcome. Thank you so much.
Madam, how to select statistical analysis tool for QBD analysis? Is there any?
So as I mentioned, there are many softwares which will auto-generate, like based on how many factors you have and how many independent and dependent variables you have. These softwares can give you a list of designs that you can utilize. But again, as I mentioned, if you are a brand company, if you have all the time and money, you can do those big designs. But if you are a generic company, usually you are time market is very short, and you want to do very fast work in the least expensive, right? You don't want to spend a lot of time into doing your optimization, and you don't want to spend a lot of money also. So you have to choose the best design which will give you the information, plus they are also satisfied that you have done some optimization study, plus looking from the company's aspect that you are not going to really waste the time and resources. So you have to balance between that to think about the best possible design.
Thank you very much, madam, for delivering such a informative talk on QBD. As a part of love from our institute and SPER for the valuable time you spared for us for delivering such a nice session. We are over here presenting certificate to deliver talk.
Thank you so much for that.
Visible, RP sir?
Yes, it's visible.
Yes, I can see it.
That is visible. Thank you very much, ma'am. Thanks for your time.
Thank you.
And
Thank you very much. Thank you.
Constantly needing your support.
Thank you. Okay, bye-bye.
Welcome, Dr. Paul.
Hello, can you all hear me?
Yes, sir. Yes, sir. You are loud and clear.
Yeah.
Most
Thank you.
welcome to this conference.
Thank you.
I am Dr. SS Puntuli.
Please are requested to mute your mics, please.
Remove this certificate and introduce next speaker.
Yes, sir. Sure, sir. SK Jain, sir.
Yeah. Am I audible?
Yeah. So today we have second chair, Dr. Sanjay Jain, sir. He is a professor of pharmaceutics and presently working in the Department of Pharmaceutical Sciences, Dr. Hari Singh Gaur, Central University, Sagar. With over 31 years of experience in teaching and research and 2 years in industry, he has more than 195 research publications with more than 55 H index and 220 I-10 with over 13,000 citation index. He has 1 US and 1 European patents to his credit and also filed 3 Indian patents. His main research area is design and development of various technology-based new drug delivery system for the treatment of disease associated with gastrointestine and brain. He has awarded prestigious and highest award, Visitors Award 2018 by His Excellency Shri Ramnath Ji Kovind in 2018. Recently, he has been ranked under 2% world scientist by Stanford University, USA. Sir has published books. He has also published various research publications under his credential. He is also and he is also referee of various national and international journals. Welcome you, sir.
Thank you. Thank you very much, Dr. Geeta. So,
excuse me, Dr. Geeta, you have to stop presenting this.
Yes, certificate.
Yeah. Namaskar, sir. Welcome to this conference.
Namaskar. Good evening, Dr. Paul.
Yeah, good evening. It's much later here.
Yeah.
Singapore.
It is afternoon in Singapore, or
now it's about almost 9 p.m.
It's a 9 p.m.
2.5 hours behind you.
Yeah. So, well, I've been told to introduce you to the our delegates. So, well, I think everyone should mute their mic. Okay. So, well, today's second speaker is a very well-known personality and scientist in the field of pharmaceutical technology. He is having very vast experience of teaching and research. For our young delegates and budding pharmacists, it is required to introduce Professor Heng to our delegates.
Please, participants, mute yourself. Host is requested to mute all participants. ओ सर को तो अनम्यूट कीजिए.
Sorry. Am I audible now?
Yes, sir, you are audible.
Right. So, well, thank you, Dr. Geeta, and giving me chance to introduce such a high personality to our delegates, Dr. Paul. And today's second speaker, as you know, is very well-known personality and scientist in the field of pharmaceutical technology. He is having very vast experience of teaching and research. For our young delegates and budding pharmacists, it is required to introduce Professor Heng to our delegates. Dr. Paul W.S. Heng obtained his basic degree in pharmacy and doctorate degree in pharmaceutical technology from National University of Singapore. He is then joined the Department of Pharmacy, National University of Singapore as a faculty member and teaches pharmaceutical technology for over 3 decades. He served as head department for 2 terms between 2000 and 2004. He is the principal investigator of GNUS Pharmaceutical Processing Research Laboratory, which is a research laboratory focused in process and product development related to pharmaceutical technology. He is currently an adjunct staff of the National University of Singapore and still runs GNUS along with his company, GNUS Technology, and a research institute in Nanjing, China. His research interest is in pharmaceutical technology, especially research related to solid dosage form, particulates, pellets, and tablets. He has expertise in design of control release system, inhalable system, encapsulation technology, along with product quality and stability testing. He has successfully supervised and co-supervised over 60 doctorate programs students, several master students, author and co-author over 30 SCI journal articles, and has written several chapters and patents. He is the editor-in-chief of Asian Journal of Pharmaceutical Sciences. He is in the editorial boards of the Pharmaceutical Development Technology Journal, Journal of Microencapsulation, Drug Development Industrial Pharmacy, Therapeutic Delivery, AAPS Pharma Scientech, Journal of Pharmaceutical Sciences, Interjournal of Pharmaceutics, and many more. Today, he will be delivering his lecture on moisture in solid dosage form, investigations and impact. Now, I invite Professor Heng for his talk. Before he start his talk, I would like to say our delegates, if they have any question, they are requested to write in chat box. So after the Professor Heng talk, we'll discuss about your queries. Thank you very much, Professor Paul W.S. Heng. Please.
Thank you very much for the introduction. And I would like to thank the SPER committee for this invitation, especially Dr. Nagesh, who has been very instrumental in my participation. When I was asked to give a talk, I thought that this was an education seminar. So I tried to get a topic which is more related to education and perhaps university research. And it's a topic I have been involved in the last, extensively in the last 10 years, which is very productive because we publish a lot of good papers in International Molecular Pharmaceutic, JPharm Science, IJP, and so on. So I think this is an area which perhaps lack attention, and especially with moisture. I think it's a simple topic, but it's something anybody can do. So I would like to share with you, because I think in India, you have a lot of good budding scientists, and I think the reach is very, very large. So I will divide my lectures into 3 parts. I'll try to do it a little bit faster because of we have a lack of time. We will talk about introduction of solid dosage form and moisture. Then we will talk about the presence of moisture, and then what are the aspects of moisture will impact excipient and dosage form. So this is just a touch, but I have a lot of papers. I think if you take my name, you Google, you will get a lot of papers on stability, which I think because this is a little bit old, I didn't want to incorporate the new topics of research because it's very complicated and the time doesn't permit good, because a lot are very equation-based development. But let's look at solid dosage form, okay? Now, as you all know, moisture is something you cannot avoid. Any dosage form will have it. So where the dosage form will come especially heavy is when you do granulating, pelletization, you do coating. But more important, moisture is in the environment. So, you know, you cannot have a super dry environment. And moisture is also important in dosage form because you need compaction strength, for example. You cannot use a bone dry material. Static will be very high. So, you know, it is a something you have to toggle with, right? Because it is innocuous, it's everywhere, it is sometimes harmful. But it's harmful if your product is moisture sensitive. But even if your product is not moisture sensitive, there are elements of moisture that can affect product, which I think is important. Let me look at how moisture works. It can affect the performance of a product, like dissolution, disintegrating, tensile strength. It also affects stability, and this is of great importance in product development, especially when you have got moisture sensitive product, and chemical stability, physical and chemical stability. And many a times, like for example, vitamin C is a good example. You know, it's a very tricky, although in terms of discoloration is not great to, in a way, it's surface, but it makes the product not acceptable, okay, because of discoloration. So let me just discuss very briefly on the presence of moisture. How do you find out, or how do you measure moisture? Now, first, I think I know many of you would have known this, but for completion, I think I should list it out. One of the ways to find moisture is by Carl Fisher. I think many of you could have used this, but I just put it there as a totality of the lecture. So you can use volumetric, colorimetric. I think most of you would have done. I will just quickly go through this because we can spend some more time with other things. But moisture measurement is important. Now, but moisture can be considered also as a volatile. If you don't have a good Carl Fisher, you can actually use an oven. Best is to use a vacuum oven. Normally what we do, we preheat the oven, we put it at 105 degrees, And once we think it's dry, we vacuum it and we just let it cool. Okay, then you get a bone dry material. It is very important to get a very dry material, because you want to study moisture, you must first start with something that is dry. Now, moisture balance is a good way. A lot of you will use it to measure moisture balance. Of course, thermometric analyzers are also another way to measure moisture in micro quantities. And this is very important when you look at hydrates and solvates and such like material. Now, for research, there are other ways by which we can measure moisture. And I think this is something that you all could think about. Are there other methods? And I just want to touch briefly on our use of dielectric spectroscopy. And dielectric spectroscopy is an interesting process which you all can read up if you're interested. There is a literature reference there. You basically look at the orientation, polarization of, but it is a little bit mathematical in looking at this, to look at the dielectric relaxation. But the principle is very basic. You actually pass electric current and put in an insulator in between, and then you measure the dielectric of the material that you put in. And by this, you can actually work out the moisture and the condition of the moisture. So the method is, this is the dielectric. Normally what we do is that we use liquid nitrogen to cool the chamber up, and then we slowly raise the chamber and then measure the dielectric with time. And that will give you a pattern of the dielectric, basically the ionization, where you get the temperature in Kelvin and the frequency. You can see that this is a native starch, and the pattern of the dielectric constant is a bit about the permissivity is a bit different. There is a little hum here that shows that there is something that is developing in this native starch. But if you use sodium starch glycolate, it's just a flattish liquid. So you know that native starch and sodium starch glycolate are different. Okay, and they have got, this will give you some idea on that the moisture present in a native starch is a bit different from the moisture in sodium starch glycolate, in the sense that there is a bit of a hum here showing that there's a captive position where the moisture is trapped, and therefore that's why you get this situation. Here you get a flattish, that means the moisture just evaporate. Now, this is just a simple way of explaining it, but of course there are a lot of explanation regard to the relaxation. Now, of course, the moisture, that means the moisture present in starch have two formulas. One is bound and one is free, okay? So you know that the bound moisture and the unbound moisture behave differently. That's why you found that the bound moisture will be more difficult to remove, and the unbound will be removed very easily. So that explains this phenomena. But if you want, you can read the paper. But I think for the most of the audience, we know that this is one method to look at moisture. Okay, so that where else you find that here we'll say that the water is perhaps just present and is very likely that is mobile, unlike the native starch, which there is a certain amount of entrapment. So you know very well that water that is in starch and that is structural, and what in sodium starch glycolate, which is we react the starch to a polymeric material, is different. So the behavior is different and the properties will be different. I will come to this property later on, but so you can use starch with dielectric and do it. But there are also other methods like NMR and other spectroscopic methods. I don't want to spend too much time with this, but there's a lot of opportunities for us to try to measure water in different methods and then derive their behavior, okay, or their characteristic in solids, okay, especially in excipient. Now, it is important because the water there can have different ease of being released, which we will talk about later, okay? Now, what are the shortcomings of moisture analysis? The general one, like for example, you use an oven or you use a moisture balance and so on. Because when you look at the moisture, especially with involving stability of moisture sensitive API and excipient is well studied, but the mechanism, how this solid water interaction, okay, is not very clear. Okay, why is it that this excipient, it makes it worse? A lot of time when you do stability studies, you say, oh, it's not stable, then you just change. But you actually don't understand the underlying mechanism, why it is less stable with this and more stable with that. And this is for research, is very useful, that students adopt inquisitive searching to understand whatever happens in the lab. Okay, then because the total moisture is not the best predictor for stability, because there are the moisture depends on the mobility of the water molecule. Okay, and that is where it is important that when you look at moisture, you should not look at total moisture, but look at what we call water activity, or the moisture that is migratable, or we call, in another term, is called fugacity of the moisture. If the fugacity is high, then that will be very detrimental. But if the moisture is trapped, even though there are a lot of moisture, it is not so bad. So measuring the amount of moisture to relate it to product stability is not a very good method. But it's more important to determine what is the water activity, or what is the fugacity of the water present. Okay, so I would like to introduce to you another concept, which is the water activity, which is fugacity related to the escaping tendency of water. Now, this is a very straightforward method. The equipment is very cheap. You can actually measure it. And basically, you measure the partial water with a partial vapor pressure. Now, of course, water being very volatile will be giving a number of 1. And what is that number relative to water? Okay, that is just the water activity. So the ratio, okay, so ratio that is close to 1, that means like water, and it's below that, then it is less. So you want a water activity is the escaping tendency, and this is what that is of importance when you talk about moisture affecting product, not the moisture content. Okay, this is something that a lot of people do not quite understand. Okay, now water activity is used especially in the food industry. Okay, a lot of times in the pharma industry, very few people look at water activity, because it is a product of food industry, because the food industry will determine the likelihood of microbial stability, and fugacity is a good prediction of it. So it is used very often on the perishability of product, and this is the kind of graph that you use. There are many methods to measure water activity. I think I don't want to spend too much time, but if you are interested, you can look up in it. And it is a good area for research, okay? Because I think there's very little persons working on this area in the pharma industry, but mainly in the food industry. But I think there's something that we could learn. In fact, most of my primary work, actually, we started from the food industry. And then I worked with Nestle's product, and that's how we originally did all our absorption, desorption isotherm with Nestle. That's how I acquired the knowledge and do it myself now. So the absorption, desorption is how you basically look at moisture content and the water activity, and you plot it up. This is the absorption, desorption isotherm. So normally that's how we do. We start from a very low moisture to a high moisture, and then we go back. So we look at the absorption, desorption isotope, and this absorption, desorption isotope is very useful to predict the movement of water and also to predict the protection that is conferred by excipient to the product. So I think I've written several papers on this. I think you all can easily look this up. Okay, now looking at water, you look at the states of water. Water can be bound or unbound. So unbound is a free water. This free water and the bound water is there, but it is not free. So you will say that this whole, when you measure moisture content, you will measure both the bound and the unbound water because you're measuring the total water. Okay, but when you are measuring water activity, you just measure the unbound water. So if you think about it, if you really want to study water activity or product stability, you should focus at the water activity, not the total moisture. Okay, because the bound water is tightly associated, it's part of the structural element of the material. Whereas the free water is free to move about and it can then cause degradation. Okay, it can very variable depending on humidity. So our focus should always be water activity and not moisture content. So why moisture is important? I think if you know a product actually experiences moisture, packaging and so on will be perhaps influenced by the presence of moisture, and the moisture the moisture will affect the product stability. And you know that different parts of the world, different countries, they have different moisture, especially Singapore, which is in the tropics. Our moisture is terribly high. On the average, we are 80–90%. Okay, but of course, you are in the temperate country, the moisture is much lower. So you know, a product that is in the tropics, for example, if you go to the southern part of India, it's very high moisture. So the kind of product protection is very different. Okay, so when you expose a product, sometimes it's stable in the temperate, but you export it to the tropics, it might not be stable. That's one reason why ICH now includes 75% humidity for stability studies. I think that is because of this. But also we must be mindful that often when we sell a product, the consumer opens up a product and then is exposed to the environment, and that product will then interact with the environment. And depending on the time frame the person used the product, the quality might be affected. Okay, so there are many roles moisture affect product. Okay, the raw material, of course, you know, it affects the flow. Okay, for the finished product, you get the physical functionality of a product. Or if you take the stability, where it's important, microbiological, physical, and chemical stability. And so there are many aspects where moisture play a part. Now, very often when we make a product, very quickly for academic interest, we must think of how do we protect against moisture. Of course, the environment, the humidity control, but there's a limit, because if you put a product to the patient, then the environment could be broken, because controlling it by like packaging, for example, is one way. But you know, packaging, there are many choices. Of course, if you use aluminum foil, it is very good, but you can't see the product. Often you want to see the product. But you know, a product that is very low, high permeability to water to low, okay? But the cost also is go the other way around. So a lot of manufacturers will try to use the lower cost packaging material, but they are highest in permeability. So this is one of the problems that come about, especially with aspirin tablet, and quite often they have to be withdrawn because of moisture in the environment. So coating a product can also protect the and the excipient, which we will discuss about. So when you talk about coating, you know there are different methods of coating. There are different coating material that you can use. I think I don't go too much in detail, but there is one option. But what I would like to also to bring up is that excipient also play a part. A lot of time people forget that there is a possibility of using excipient to protect against moisture. So by judicious choice of excipient, you can actually promote or build a product that is more stable. That means if incidentally the product come in contact with moisture, there is protection conferred by the excipient. And I think this is an area of my research, of much recent time, and I'd like to share with you. But of course, I cannot in this time frame to share everything. But if you look at a lot of my papers, I think you can find a lot of different avenues how excipient affect the product stability. Now, of course, in literature, if you look, people, for example, use light absorbing excipients for certain like difendipine or silica to enhance the stability of aspirin, or maltodextrin for ascorbic acid, and dextrin, Pulan for various type of products. So there are many examples excipient help in formulation, but often the underlying mechanism why it protect is not very clear. And this is where, as a researcher, it's a good area to look at and to study how it affects moisture and how it product protects a drug from degradation. So, I mean, I just give you a simple chart to show you. For example, you can see these are different type of, these are native starches, these are pre-gelatinized starches, and these are microcrystalline cellulose, okay? If you look at the degradation, microcrystalline cellulose is the highest, okay? These are degradation, you get very high. But the starches and pre-gelatinized is lowest. But if you measure the moisture content, surprisingly, the starches have very high moisture content, but microcrystalline cellulose very low. So if you think about it, microcrystalline cellulose has low moisture content, but degradation is high. So it doesn't gel, right? Because if you were to just measure total moisture content, you'll find that microcrystalline cellulose has much lower moisture content than the starches. But the starches gives a far better protection because it's less degradation. So this interesting research, you can show that there is no correlation between moisture content and the degradation rate. But if you were to plot water activity versus the stability, now the trend is much more apparent. You can see the starches have much lower water activity, and the microcrystalline cellulose is much higher. So I hope you are convinced that water activity is more important than moisture content when you want to predict the stability of a drug in the excipient, with the excipient. So I think this is just a take-home message that if you are interested in this field, this is very important and useful information. Now, so therefore, we can now know that there are such things as moisture scavenging excipient, which we can look at. So, for example, if you have got like starch, for example, it can entrap water. So it can hold high water, but it doesn't let the water run about. It actually prevents the water from, it binds water, because starches are formed like onions. I think if you understand starches, they are like onion skin, and they are amorphous crystalline material, and water, once it gets in, gets trapped inside. So therefore, the water is not free to run about. And therefore, the starch, for example, can protect or can absorb moisture and reduce the moisture activity or water activity, and therefore protect degradation of a product. So this is just a simple example. Okay, now I more or less finished my lecture, but I would just want to show to you maybe very quickly how some of the research work. Just a snapshot. I actually have a lot of it, but just a snapshot, the simple one, which I think some of you will maybe you can think about in your research. We look at tablets sensitivity to different moisture. For example, you know, aspirin, acetylsalicylic acid, very easy. Break with water, it will break down. Okay, so you can very quickly, in fact, in less than a month, you can do a stability study using aspirin. Okay, but you actually have to increase the alkalinity. You make it the pH high, so the degradation very fast, and you can finish the experiment in 2 to 3 weeks. Because it's very in alkaline condition, aspirin will degrade very fast. So you can do your experiment very fast. Okay, so I just give you a sample. I don't want to spend too much time. Excipient that commonly lactose, starch, mannitol, and so on. But you can see we make all these product, and then we study the store in under different RH condition, and then we analyze the stability. This is a very simple experiment, but you can yield very good understanding of excipient and product stability. So you can see, I don't want to spend, the moisture will have different product with different excipient. For example, the tensile strength degradation could be very different. For example, sorbitol is very bad, especially high RH. You can see it's virtually, they don't have tensile strength. Okay. And some material like lactose or sucrose don't get so badly affected. Starch is quite bad. So different excipient will have different property. The drug stability, on the other hand, is a little bit different. You can see the drug stability gets very high generally, but depending on the excipient, you get different result. But I don't want to go into too much detail because each of them got explanation, and I've written papers on them. So I think you can maybe search and look it up. But I just want to tell you that the different excipient got different outcomes. Some are more, some are less. So obviously, if you look at product stability, for example, I at 75% RH, which you can see, for example, this aspirin tablet, there is salicylic acid on the surface. You see, this is the crystal on the tablet. You can see this green line. This is a crystal. We use Raman. We can actually determine what it is. And you see salicylic is like that, whereas aspirin is a bit different. So you know very well that these crystals are salicylic acid. Okay, they're present. So you know different kind of excipient you can. Of course, sorbitol also causes deliquescence and it will affect the stability. Okay, so I think different kind of this will affect the sublimation of salicylic acid with sorbitol. So I think we can look at the stability very often to look at the product and the storage condition. Okay. Now, the sensitivity of excipient also is sensitive to moisture. Not only we talk about API, but you also remember that excipient itself, when exposed to moisture, can also vary in their functionality. So, you know, we look at like cross-povidone and dicalcium phosphate, we make tablets and we look at the property. You know, very often you can see the different RH, a different amount of excipient, the disintegration time will change. And because of RH, so it actually drops. So that is also another issue that you could see that some excipient are more sensitive to moisture than others. Okay. And they can affect the functionality of the dosage form. So the tensile strength, for example, can be very bad when you have got a high RH. So because the tablet actually can, to some extent with moisture, can change the property. So exposure to moisture cannot be often tolerated by some tablets, depending. So if low RH will be okay, but high RH can be a problem for some product. So you can see when the RH, the tablet become a bit molten, okay, when they are very high. So you know the appearance can change and the functionality also can change. So we know that the chemically similar excipient can have different physicochemical reactivity. So excipient needs study, especially we want to do substitution, product stability can be affected. Storage condition also can be of importance. I think this is well known in literature, but I just want to emphasize to some of you, especially when you have got difficulty in storage. Now, drug stability with moisture also is important. You know very well, when you do granulation and so on, it will affect moisture. Okay, so you know, different excipient can have different excipient will have different stability when you store in different RH. Now, what I want to show to you in this graph, actually, the reason why I'm showing it to understand moisture and the water activity. Now, these are just the degradation of aspirin. Okay, if you were to just look at moisture content, you see there's no correlation between moisture content. The R is very low. There's not much correlation between moisture content and the degradation. Okay, this is the degradation. But if you were to look at water activity, you see there's good correlation. Okay, the low is 0.7, but most of them are 0.9. So again, I want to attest to you about the importance of water activity and not moisture content when you want to look at stability of a drug. Okay, that is my emphasis in this lecture, so that you will go back, you understand the importance or the difference between moisture content and water activity. So for drug stability, look at water activity, not look at moisture content. Okay, I think a lot of people talk about moisture content, but they don't talk about water activity. But this is very important. Okay, I think you've got to take into context that moisture content takes into account both bound and unbound water, whereas the water activity is just a free, fugacious water. Okay, the sensitivity is more towards the water activity to degradation. Okay, I think that's what all I have to conclude. The moisture affects the dosage form in terms of drug stability as well as their performance in dosage form. Okay, excipient may be chemically similar, but their physico-chemical reactivity may differ markedly because of the way they interact with water. Product stability can be affected by the choice of excipients. Hence, excipient selection or substitution warrants careful product stability study. Even different starches, we all have shown to have different properties, because the ratio of the amylose and mylopectin can affect drug stability. Okay, because you can say starches, even different maize starch have different properties. We have seen, we have noticed. Okay, storage condition or excipients are important to preserve the excipient's performance. Because a lot of time people talk about drug, they don't talk about the excipient, but the excipient is also important. Water activity shows promise in explaining the role of moisture affecting drug stability of solid dosage form, and hence is a better predictor of product stability. I think with that, I would like to thank you all for your attention. I think I just take the quote from Wolfgang, because he is a Michelin chef. He says, Italian food is all about ingredient. It is not fussy and it's not fancy. So the kind of ingredient, the kind of excipient is about what kind of product you make. Okay, thank you very much. I am open to questions. Thank you.
Dr. Heng.
Yeah.
You have explained very nicely the role of moisture and water activity.
Yes, yes.
In the formulation development, right? It's a very vital role. Say, you said that moisture content is a good concept that you have given. Moisture content is having the lesser low as compared to the water activity. We must have to measure water activity.
Correct.
Right? Say, if you want to predict the stability, or if you want to see the degradability of the product. So you have very well explained the concept of the moisture content in the formulation development, its stability, the methods used for measuring the moisture content, apart from the Carl Fisher you have mentioned, you have explained various method, and the bound and bound water and free water, the role of these waters in the stability is very nicely explained. I think our delegates are very well now aware about the role of moisture in the formulation development. So there are only few queries, not question, it's a queries, doubt you can say, right? Your lecture is very well taken up by our delegates. And one delegate, Dr. Vijay Mishra, is keen to know about is there any vital role of water of crystallization in the formulation development?
Okay. Water crystallization is normally water crystallization generally is not active because it is a part of the structural water. Usually like lactose, for example, the lactose monohydrate we use, it doesn't affect because the lactose, the water there is a part of a structure, it doesn't move away, it's not fugacious, it is bound. So what is important when you want to measure moisture is to make sure that you know what is the free water. So normally it's interesting because you ask this question because, for example, when we are doing lactose, if you dry lactose, water of crystallization of lactose will be released when the temperature reach about 80, 90 degrees. So if you use you want to take the moisture, for example, you will be determining the free and the structural water. Okay, so what we do is that we normally heat it about 60 degrees and then we vacuum it to remove the free water but keep the moisture of crystallization intact so that we can differentiate. I mean, this is very important for students who don't understand because the water crystallization is structural water. It doesn't really affect product. Okay, but if a water crystallization can predispose it, if, for example, you heat the product, for example, depending on what temperature it is released. Otherwise, the water will not be released. But okay, I think water crystallization is interesting because sometimes when you do absorption desorption isotope, you find that the water crystallization at high humidity can sometimes affect the reading. It gets a bit complicated, but in general, I think I don't want to spend too much time describing all the little fine details, but there are pros and cons. But it's a good question. But normally, at room temperature, the water crystallization is very stable. Okay, it doesn't really affect the product.
But Dr. Paul, this is from my side, or you can say it's my experience when I was in one pharma industry.
Yeah.
And there was a question that why you are not using magnesium stearate as a lubricant, right? And especially for the manufacturing of this aspirin tablet. And they said that it is because water of hydration is 2 or 3 molecules are there. That is why they are not using, because it will hydrolyze the aspirin. So they were not using magnesium stearate.
Okay.
And yes, please.
Yeah. Go ahead. I go ahead. Okay. It's very interesting that you know because I study magnesium stearate quite a lot, and generally, also I work with FASI, and we are now characterizing 15 types of magnesium stearate from them. We also do a lot of, we work with some professors who make crystal. We study using TGA, we actually work at how the hydrates, etc. Magnesium stearate is a hydrophobic compound, actually. There are moisture content, but you know, the magnesium stearate water is a bit tricky. I do not know why. I'm not quite clear, but maybe they don't love water that much. That's why sometimes.
Yeah, that is correct. That is correct, Dr. Paul. It is a hydrophobic in nature, but it is same time it is having 2 molecules of water of hydration. And that too molecules of hydration, because it is different from water of crystallization.
Yeah.
Water of hydration is affecting the stability of the
Correct.
product,
Correct.
API,
Correct.
not
Yeah.
the water of crystallization.
Correct.
And
So,
you said very correctly the unbound water and bound water.
Yeah.
The bound water is generally does not affect the stability. What
Correct.
I gather from your lecture,
Yeah.
and the unbound water is having the much effect
Correct.
on the stability of the product.
Correct.
And this could be one of the reason, but you use silica, 3% silica for enhancing the stability of the SP.
Not me, it is literature.
Achha, it is literature. Okay, okay. So in the literature, it is given 3% silica is used for enhancing the stability. All right, that is maybe degradation. You are preventing the degradation of salicylic acid by using the 3% silica. But the same time, if you use 3% silica, will not it affect its disintegration time and dissolution time, because it will make a hydrophobic coat over the surface of the particles of the salicylic acid, doesn't it?
Yeah, but you know, silica is a very interesting product. We can spend a lot of time talking about silica because I worked with silica for so many years. From the day when they put silicified microcrystalline cellulose, I was involved to. Now, silica is interesting. Also, there are different types of silica. There are many types of silica. You cannot grade silica all the same. Some silica are porous, some silica are less porous. And you can actually find silica of different porosity. Okay, it's very interesting. Now, I think the stability in the case of the literature paper, I think silica, you must remember, once you put silica like 3%, it actually causes a physical barrier between materials. It actually, like, you know, is like a coating, a powder coating of the material. It separates material, and this can enhance stability by physical separation. The other reason also silica, it is not true that silica actually decrease solubility or what. Silica sometimes is a great product to make certain product. For example, if you are making herbal extract product, they are very, very hygroscopic. Okay, silica is great. We use silica to enable this type of product to disintegrate. Actually what it does is that it slows down the gelling of hydrophilic material, the gelatinous material. If gelatinous material get water, they will gel up and the whole tablet become a lump. So to prevent that, you put silica. Silica will actually coat the gelatinous material. So when it goes to water, you're quite right. It actually help to separate the material without them gelling up, and you enable disintegration. So people who cannot disintegrate a herbal product, silica is a great product. But always remember, silica is good in a way, but the tool maker also loves it because you wear down your tool very fast.
Very nicely said.
The tool maker loves silica because silica is very good for grinding up your punches.
Yes. Very nicely explained it. Say one more query from my side. It is from my side. Is there any effect of this moisture content on the flowability of the granules from tablet punching machine from hopper to the die cavity? And up to what % of moisture content will affect this flowability?
That's a good question. Moisture, okay, how moisture affect material flowability? Granules are quite large. Generally, it is not going to be a problem. Generally, but it depends on the moisture of the hydrophilicity of the material. If you have a material like that loves moisture, then in the presence of moisture, they will absorb. Even at 50% humidity, it will start absorbing, and then it becomes sticky. The problem is that stickiness is not, then this becomes a problem. So normally, some product we have to maybe reduce the ambient humidity to 20% moisture because it's very hygroscopic. Okay, occasionally you have this kind of product. But in general, generally at about 50%, most product will not, the moisture will not affect flow very much. But if you have a product that contains a hydrophilic material, like low molecular weight hydrophilic material, then you have a problem. You will absorb moisture very rapidly, and the moisture will be absorbed on the surface and makes the product sticky. Now, you might not affect flow per se, but it will affect the quality of flow. Then if flow is erratic, then you got a problem in weight variation. So I think one has to be mindful about that. So here what we do is that we try to reduce the humidity of the chamber or the room by with a dehumidifier. For example, a good example is that when I think we tend to have a lot of this problem when we are looking at food industry. When we are like dealing with seafood, spray dried seafood is very, very hygroscopic. So what we do is that we actually have a curtain of dry air to actually reduce the moisture, because the material, once they take moisture, become very sticky, and the whole powder just stop flowing. Right. That is a. But in general, for pharma, this is rare. Okay, very rare. Unless it's a badly. But if you work with herbal product, there may be a problem. Herbal product, especially extracts, you know, where they are very problematical. I think those of you who will work with extracts, you understand this. But for pharma, the drug product, generally these are rare. But of course, the moisture will also impact another factor, is sticking problem. You know, if you have a product that is not so much affect flow, it affects sticking, and then the punches get sticky, and then picking and sticking occur, and that can be a bit of problem. And then you probably have to clean your tablet press quite often, which is going to be a nuisance actually.
Thank you, Dr. Paul. Is there any queries from the delegates? Otherwise, we can shift to Dr. Geeta and Dr. Pancholi. Dr. Paul has explained very nicely. Geeta, please unmute your mic first.
Sorry, sir.
Yeah.
Thank you very much, sir, for inviting our invitation.
Thank you.
And delivering such a nice talk and giving a detailed, updated information about how the moisture will affect the formulation, as well as the different ingredients can be affected, excipients can be affected by moisture, and degradation studies can be there, which is affected by the moisture. Thank you very much, sir, SK Jain sir, for handling session very nicely. So as a part of token of appreciation on behalf of my institute and SPER, I want to present certificate for delivering your session.
Thank you very much for the honor. It's indeed a very nice to be able to share with you some of our research work, which we work with a lot of passion. I know all of you professors, you're very passionate about the research and very proud of your work. So am I. But I am actually on the way out of academia.
You are associated 10 or 20, 15 years with the academic institute. You are more academician than the scientist.
But
you are teaching since
yeah,
last
but,
10, 15 years.
30 years, over 30 years.
30 years. Oh, such a nice experience. Yeah. So you are the professor of the professor. Thank you, Dr. Paul. Thank you very much.
Thank you.
Yeah, thank you both Professor Jain and Dr. Paul.
It has been very nice talk, and really I am very hopeful that all the delegates enjoyed this learning session. It is a practical aspect of formulation. Everybody needs to know, and many times I would say even the teachers, they are not emphasizing on.
Sir, your mic is mute. Pancholi sir.
Pancholi sir, your mic is mute. Please unmute it.
Thank you all, sir. Thank you very much.
Thank you. Thank you very
Thank
much.
you, sir. Thank you. Thank you all. Have a good day.
Yeah, thank you.
Okay.